Good morning, sir.
Yes, sir. Good morning.
Yes.
Hi, sir. How are you?
Fine, sir. Fine.
Yeah.
Whether my audio is clear, sir?
Yeah, your voice is clear, sir. No problem.
Sir, if you're free, can we test the screen sharing once, just for 1 second, whether it is visible or not? Can we see, sir? Hello, sir.
Yes, sir.
A few points.
Can we test the screen sharing once, sir, whether it is visible or not? For the...
You can, sir. You can.
Okay.
మా టీచింగ్ దాంట్లో ఆ లింక్ అదే మరి సేమ్ ఓకే నేను ఎలా మరి స్టార్ట్ చేసేస్తారా yes మేడం రాజీవ్ సార్ ఒక్కసారి చెప్పేసి స్టార్ట్
Hello.
Sir, is it visible, sir? Sir, is it visible?
Yes, it is visible, sir.
yes sir it is visible sir
Okay, thank you.
Sir, good morning.
Good morning.
Sir, shall we start the session?
Okay, no issue. You can.
Good morning, Dahiya sir.
Good morning, Satish sir. How are you?
Fine, sir. Fine, sir. Hope you're fine.
Welcome.
Hello, Suresh sir, are you there?
I am here only.
Okay. Actually, your video is turned off so that we are not recognized you.
Okay, no problem. I am here only, no problem. Carry on.
Okay, thank you, sir. Thank you, sir. Okay, shall we start?
Good morning, ladies and gentlemen. A very warm welcome to all the dignitaries, participants, delegates, and dear students. A warm welcome to the second day of 17th Indo-US Virtual International Conference with the theme Advances in Global Trends in Pharmaceutical Sciences, organized and hosted by St. Paul's College of Pharmacy, Turkayamjal, Hyderabad, in collaboration with APP Karnataka State Branch, APP American International Branch, and Analytical Chemistry Division. Hope you have witnessed the wonderful sessions yesterday, and now I suggest you to get ready for the power-packed sessions from the most renowned speakers. We are honored to have with us today the Honorable Guest of Honor, Dr. Rajiv Dahiya, sir. Good morning, sir, and welcome to the session.
Thank you.
Dr. Rajiv Dahiya is Doctor of Science in Clinical Pharmacology from International University for Complementary Medicine, Colombo, Sri Lanka, and Doctor of Philosophy in Pharmacy by Uttar Pradesh Technical University, Lucknow, India, for the thesis entitled Studies on Bioactive Cyclopeptides and Peptide Derivatives of Heterocyclic and Other Aromatic Compounds in December 2008. Dr. Dahiya is presently Director, School of Pharmacy, Faculty of Medical Sciences, the University of the West Indies, St. Augustine, Trinidad and Tobago since February 2016. He is President of Association of Pharmacy Professionals and Editor-in-Chief of reputed pharmaceutical journal of international level, Bulletin of Pharmaceutical Research. Until now, Sir has published more than 75 research papers and review articles in various peer-reviewed international and national journals with impact factors and 8 book chapters with Elsevier, Springer and Taylor and Francis and Australian innovation. And Sir is having 1 Australian innovation patent. He has been invited 26 times as a guest speaker in various conferences, including 39 orations and 109 poster presentations. Dr. Dahiya is honored with Most Productive Researcher Award in March 2019 on occasion of FMS Research Day by Dean Faculty of Medical Sciences. Sir is also felicitated with Caribbean Distinguished Pharmacy Research Award in August 2021. Dr. Dahiya has supervised more than 20 MPharm projects and 4 PhD projects. He is reviewer for various Q1, Q2 best quartile journals with good impact factors. Sir has research indices of 24 H index in Google Scholar, 19 in Scopus and 17 in Web of Science. With this brief introduction, I request Dr. Dahiya sir to please address the session. Over to you, sir.
Good morning, all. So, I welcome all of you on the second day of the 17th Indo-US Virtual International Conference hosted by St. Paul College of Pharmacy on the theme Recent Advances and Global Trends in Pharmaceutical Sciences. As yesterday we discussed, so this conference is particularly organized in commemoration of Parakram Divas, which is related to Netaji Subhash Chandra Bose, and we salute his contribution made by him toward the freedom of the India, and we remember his works, hard works on this occasion. So, briefly summarize, yesterday there were sessions from Professor Somnath Singh, who belongs to Creighton University, Omaha, Nebraska, and who delivered a lecture on genomic revolution in pharmacy practice and drug development. It was followed by another interesting lecture by Dr. Sivkanya Filoria from AIMST University, Bidong, Kedah, Malaysia, on the topic technology-based citation and referencing tools. We also heard from the Vice Chancellor, Sabarmati University, Ahmedabad, Gujarat, Professor Madhavbhai M. Patel about the latest development in pharmaceutical research, as well as there was an addressing by the American International Branch President, Professor Alekha Das, who is Associate Dean of Research at Department of Pharmacy Sciences at Creighton University, Omaha. In addition to that, we organized a oral and poster session, and along with that, there was a interesting oral competition of 7, composing of 7 orations from different states of India, and it remained very fruitful, and discussion will benefit our young bodies. Today, is the second day, and it will continue with the scientific lectures of 3 eminent speakers. First one being Professor Kuntal Das, who is our president of APP Karnataka State Branch and a professor at Kripanidhi College of Pharmacy, Bangalore, Karnataka. He will deliver on application of additive manufacturing technology in vitro conjured Indian threatened medicinal plants for isolation of bioactive compounds in healthcare management. He is a pharmacognosy expert.
Good morning,
it will
sir.
be followed. Good morning, sir. And it will be followed by the lecture of Professor S. Satish Kumar, who is Professor of Pharmaceutics at Wells deemed to be University Vistas, Pallavaram, Chennai, who will deliver on nanoparticles, a successful tool for delivery of antimicrobials across BBB, blood-brain barrier. And the last lecture will be by Professor A. Sridevi, who is Professor of Pharmaceutical Chemistry at Sri Padmavati Mahila Viswavidyalaya, Tirupati, Andhra Pradesh. She will deliver on phytochemical and pharmacological evaluation of roots of Jasminum auriculatum for nephroprotective activity. So these 3 lectures, and along with that, there will be oral and poster presentation continuation followed by the valedictory function. So by taking not much time, I would switch over to the anchor so that the respective scientific session of today can be started. Thank you very much for giving me opportunity to speak, and I keep the scientific session open with this. Thank you.
Thank you so much, sir. Thank you so much for your brief note. And for today's scientific session, so we have the chairpersons, Dr. Chennupati Venkata Suresh sir, Dr. M. Kiranmai madam, and Dr. P. Sunil Kumar Chaitanya sir. Dr. Chennupati Venkat Suresh sir is presently serving as professor and head of the Department of Pharmaceutical Analysis at Nalanda College of Pharmacy, Nalgonda, Telangana State. In the past, he served as principal at Green Loyal Academy of Pharmaceutical Education and Sciences and Mother Teresa Pharmacy College. Dr. Chennupati was a recipient of Bharat Sikha Ratna Award on the occasion of National Seminar on Individual Achievements for Economic and Social Development at Constitution Club of India, New Delhi on 30th September 2013. Dr. Chennupati received APP Young Performer Award on 18 January 2014 during APP 3rd Annual National Convention on Novel Drug Discovery and Development. Current status and challenges ahead, organized by APP in collaboration with the Faculty of Pharmacy in Gujarat State. Dr. Chennupati was honored with India International Achievers Award for Educational Excellence by Global Society for Health and Educational Growth. Dr. Chennupati published one book entitled Pharmaceutical Inorganic Chemistry by PharmaMed Press. Dr. Chennupati is a life member of various professional bodies like Association of Pharmaceutical Teachers of India, Indian Pharmaceutical Association, Indian Council of Chemists, Association of Biotechnology and Pharmacy, Indian Hospital Pharmacists Association, and many other more. Dr. Chennupati was a convener, co-convener, chief guest, guest of honor, speaker for more than 50 national and international conferences held at different places at India and abroad. Dr. Chennupati published more than 15 research and review articles in peer-reviewed national and international journals indexed with the Scopus and Good Impact Factor. We welcome you to the session, sir.
Thank you, ma'am.
Thank you, sir. It's my privilege to introduce our another chairperson, Dr. M. Kiranmai, presently working as a professor and vice principal at St. Paul's College of Pharmacy, Hyderabad. Madam received Doctor of Philosophy in Pharmaceutical Sciences from JNTU, Hyderabad in 2014. Madam is having 18 plus years of teaching and research experience in various pharmacy colleges and chairperson for scientific session Percept OU. Madam has published various publications in various international and national journals and also presented papers in national and international conferences. Madam expertise research areas are medicinal and green chemistry, nanomedicines, and multidisciplinary research. Madam has excellent people management skills, and Madam has been as a guest speaker in the International Online Scientific Congress, Phytomed 2020, organized in Melbourne, Australia. Madam has almost 53 international and national publications, 55 international or national conferences, and 478 citations with H index of 11 and ITIN index of 11. With this brief note, Madam, I welcome you to today's scientific session. Welcome you, Madam. I'm honored to introduce the chairperson, Dr. Sunil Kumar Chaitanya, presently working as a professor and HOD Pharmaceutical Analysis at St. Paul's College of Pharmacy, Hyderabad. Sir is graduated in B.Pharmacy in 2002, M.Pharmacy in 2005 from Mangalore, NGSMIPS from Rajiv Gandhi University and received Doctor of Philosophy in Pharmaceutical Sciences from Rajiv Gandhi University in the year 2011. Sir is having almost 16 plus years of teaching and research experience. Sir has published various publications in national and international journals and also presented the papers in various conferences. Sir has 32 papers published and 10 conferences presented with a citations of 232, H index of 9 and I-10 index of 9. With this brief note, I welcome Dr. Sunil Chaitanya sir for today's scientific session. Welcome you, sir. With a brief introduction of the chairpersons, now it is my privilege to introduce our today's guest speaker, Professor Kuntal Das sir. Dr. Kuntal Das sir is working in Krupanidhi College of Pharmacy, Bangalore, India. He is having total 18 years of teaching and research experience. He is specialized in the area of cultivation and standardization of medicinal plants, estimation of phytoconstituents through various instrumental methods, tissue culture of endangered plants, pharmacological activities of plant extracts, formulation and clinical investigation of plant products. He is active BPharm and MPharm examiner, PhD guide with thesis validator, paper setter for BPharm and MPharm validator, project evaluator for various universities. His career reflected with various prestigious awards and acclaims in many scientific forums. He is recipient of START Award 2021, Outstanding Scientific Award 2021, Best Scientific Award 2020, Best Professor in Pharmacognosy and Natural Product Chemistry 2019, Fellow Award 2019, Best Scientist Award 2019, Pharmacy Teacher of the Year 2018 by APTI Karnataka Branch, Fellow of Indian Chemical Society 2018, Fellow of Academy for Environment and Life Sciences 2018, Best Scientific Award 2018, and many more. Sir is having 17 years of research experience. He has guided 20 master's students and presently guiding one PhD student. He has research collaboration with many countries, went to Romania, Austria, Netherlands, Belgium, and Thailand for collaborative research and received traveling expenses from DST India. He received 9.4 lakh research grants from RGUHS and VGST Karnataka, and another 9.54 lakh grant is in final stage of approval. He has more than 112 scientific publications in various peer-reviewed national and international journals of repute. He has contributed 8 book chapters and 13 books in his expertise area. He is serving as editorial board and reviewer committee member for more than 19 peer-reviewed and national and international journals. With this brief note, I invite Dr. Kuntal Das sir to please take over the session. Over to you, sir.
Very good morning, madam, for giving me the brief introduction about myself. So I'm just telling to everyone is a very happy and safe new year. All of you be healthy and safe. So today's session, I'll start with my topics entitled. So is it visible?
Yes, sir, it is visible, sir.
Right.
Sir,
So the entitle of my today's lecture, the application of additive manufacturing technology in vitro conserved Indian threatened medicinal plants for isolation of bioactive compounds in healthcare management. So this is the title why I have taken, because this is most important for the researcher as well as MPharm students who are all doing the research for any pharmacology aspect or for pharmacognosy aspect. It is very helpful for them to select the plant as well as they can select the different type of the research from this my lecture. So let's start with my first slide. So the main thing is whatever the activity we are getting from the plant, it is mainly based on the phytoconstituent or the bioactive compound. So you can see that about 4.4 billion people of the developing countries, when they are mainly depends on the traditional medicine, and you can say about 80 to 83% of the world inhabitants who are rely mainly on the traditional medicine for their pharmacy healthcare, and they are mainly using for the any kind of the preliminary health care. So the main is nothing but the phytoconstituent or the bioactive compound which are responsible for the protecting the plant against the many environmental threats. They are known as the secondary metabolites, and various type of the phytoconstituents, mainly you can say the glycoside, alkaloids, flavonoids, tannins, xanthone, terpenoids, quinone. So these are all the most essential bioactive compounds which are available in the plant. So all the plant constituents having their specific action. So whoever doing the pharmacology research, they may know better than me. So all the constituents, mainly the alkaloids, glycoside, then tannins, resins, flavonoids. So they are having their own specific activity. I will not go for the details of the study. Just I have highlighted all the points here as per the different type of the bioactive compounds with their respective activity. The main importance is the functional bioactive compounds. It plays the major role in the treatment of the disease because of their chemical nature, and these chemical compounds possess the enormous structural and chemical diversity because of their geographical location, cultural condition, so soil nature, so climatic conditions. So all these things having the impact on the plant to get the bioactive compound in a different ratio and different kind of the chemical compounds. So various kind of the bioactive compounds, it is act as a lead compound for the molecular targets for the range of the life-threatening disease, including the cancer, diabetes, and the neurodegenerative diseases. So various kind of the treatment we are going for using the various kind of the bioactive plant constituent, which is the lead compound of the isolated compound. So nowadays the focus is on the isolated compound from the plant originated, and they are showing the more activity for the therapeutic benefits and all. So development of the new drug is a complex method. It is also the time consuming and also the expensive process. So it is the, you can see the flowchart what I have given. It is very simple flowchart and it is easily available. So it is the medicinal plants you are taking, and you are going for the first physicochemical analysis. You can go for the chemical test, and you can go for the phytochemical screening. Then we are going for the different type of the extract preparation, going for the bioassay, and we are going for the fractionation process for the isolation of the pure compound. And side by side, you can go for the study of the toxicology to find out any toxicity of the plant is available or not. And when you are going for the bioassay and all, you are isolating the pure compound. So this pure compound also you are going for the characterization, and we are going for the structural modification. And if it is the new compound, then we are going for the various type of the synthesis as well as derivatives for getting the higher potent activity. So this is the latest flowchart for the plant, how we are going to get the bioactive compounds from the plant sources. Now you can see some steps involved for the lead development. So this is most important things, any kind of the new drug development, we are having some series of the process. So you can see here the phytochemicals we are getting from the plants or bioactive compounds. First step, we are going for the discovery, and we are going for the various type of the screening technology, and we are identifying the lead compound. Then we have to go for the lead optimization by using the various structure of the chemical structure and based drug design method. After that, we are going for the pharmacokinetic study in the step 3, where the absorption, distribution, metabolism, so all this toxicity study you are studying, and we are developing the lead. After that, we are going for the clinical trial, which is most important nowadays. So first we are going for the preclinical study. After that, we are going for the human trial. So in the preclinical study, we are going for the animal experimentation, and in the higher clinical study, we are going for the human subject for the clinical trial in the different type of the phases. So you know very well, we are having the phase 1, phase 2, phase 3, and phase 4, and the post-marketing. So after that, if it is accepted, then the FDA will give the approval and will get the establish the bioactive compound as a drug molecule, potent drug molecule. So this is the steps involved for the lead development from the plant bioactive compound. Now I will show you some compounds which are bioactive compounds which is easily available as a lead compound from the natural sweeteners. You can see here glycyrrhizin. It is obtained from the Glycyrrhiza glabra. It is approved by the country Japan. Then steviocyde, ibudeside, it is very famous drug as an antidiabetic, which is obtained from the Stevia lebudiana. Then margocide, it is also given approval from the Japan. It is a Serratia grosservani. Then phyllodoxygen, which is also it is obtained from the Hydregia macrophylla. Then thumatin, it is also obtained from the Thaumatococcus danili. So this is the different type of the bioactive compounds, either natural sweeteners. This is given the approval from the different countries from the originated as per the bioactive compound. You can see here the pie chart where the plant sciences, you can see 18% research is going on, and the pharmacological pharmacy in the research we are having 21%. So rest on we are having the biochemistry, molecular biology where 15%, agriculture having the 13%, and we are having the rest. So the plant science and the pharmacological aspect, it is having the lot of scope in the future and coming year. So there are a lot of demand is happening due to the less side effect as well as having some sort of the bioactive compound to isolating and all these things. So we are going for the different type of the technology, and by this technology we are developing various kind of the bioactive compound which is not at all possible in the earlier stage. So this is the basic things. Now it is improving, and based on that we can see here I've given some sort of the histogram graph where you can see the origin of the new lead compound from the natural sources. So in the plant-based drug, you can see in the 2019 it is reduced from the 2001. It is having the 2001 having 61.5, whereas in the 2010 it was highly picked up in 61.7, and it is 2019 it is reduced in 61%. So why it is happening this one? Because the plant is getting the rare, they are getting the threatened, they are getting endangered because of the population growth and over exploitation. So based on that, the we are not getting the raw material for the development of the any bioactive compounds from the plant sources. So that is the graph is showing the declination of the bioactive compound isolating from the plant. And same thing we are having for the other sources like marine sources, microbial sources, where the terrestrial animals as well as the unspecified organisms. So these are all showing some improvement, whereas from the plant-based we are getting the less. So you can see here, as per the plant species International Union for Conservation of Nature, it is given 2018. So as per their data, the September 2019, they have listed around 2,597 critical endangered plant species. So in that things, we are having, they are also told that 21,882 extant species, and they are evaluated, and in that 20,708 of the species fully assessed, and 11,577 to 13,451 are threatened medicinal plant, whereas 189 to 352 are the extinct in the wild. So this is the data they have given, and you can see the population of the plant sources of the forest in India, how it is day by day decreasing. So you can see there is a Western Ghat region and there is a Eastern Ghat region. Apart from that, we are having in the north side where the Uttarakhand and Himachal Pradesh, they are the hub of the medicinal plants, but it is day by day it is decreasing due to the overpopulation and due to the habitat loss. So that's why there is a requirement for the conservation of the medicinal plant, and we have to go for the higher study. So you can see here from the Western Ghat region, among 5,500 flowering plants reported from the region, in the Western Ghat region, and 2,015 species are reported as endemic, whereas in the Western Ghat region, the southern Western Ghat region containing the southern part of the Karnataka, Kerala, and southern Tamil Nadu are the richest in the terms of the diversity, because they are the range of the Western Ghat region. So we are getting a lot of medicinal plants from this area, and the plant diversity in the area estimated over 4,500 flowering plants, in which the around 1,500 are the Western Ghat endemics. So this is the data. It is given by the IUCN. Then in the northeast region, where we are having the Mizoram, Tripura, Nagaland, that area. So in that area, the total medicinal plant or the forest zone around 2,74,680 square kilometer. The area is a part of the global biodiversity hotspot, and in that the total around 3,488 flowering plant species have been reported from this state, in which the 1,836 species are endemic and 924 species are having the medicinal importance. So you can see so much the vast medicinal plant sources are there in India. It is still we are not knowing at all what are the application and what is the bioactive compounds. So these are all some sort of the rare plant species. So who are all taking the research project from the pharmacology or the pharmacognosy department. So it is very helpful for them to identify, to select the sources of the raw material, so that it will be easy for you to go for the patent as well as for go for the any kind of the higher publication. So you can see here, I've given some examples from the biological source family and where the plant is getting rare. So these are all the regions. In that region, the plant is getting rare plant, and you can see the diagram. It is very nice diagram for the all the plant sources. You can see the Polygala irregulatus, then Lotus corniculatus, then Silotum nudum. Okay, so then Abitulum indicum. So these are all the various type of the species. Already it was there in India, but now it is a rare plant because of their habitat loss. So these are all some examples, and you can see some threatened medicinal plant. So here we are having so many plant species. So these are all the plants where in this region, nothing but the Andhra Pradesh, Tamil Nadu, Kashmir, Karnataka. So many plants. It is now very threatened plant. They are becoming the endangered, and they are above to the list of the red level. So as per the IUCN. So you can see here the plant photo. So where the diagram you can see here, it is look like the Amentotaxus asemica. So this is look like your Taxus plant. So but there is a little difference between the species, where the leaf of this plant is little bit broad than the Taxus plant, but it is look like similar things. Then we are having the Pterospermum reticulatum, where the leaves and flowers are more important, where the Diosporus celebrica, here the roots are and fruits are very important for the therapeutic activity, whereas you can see in this plant where having the flowers are most important. Then we are having some sort of the endangered plant species in India. So mainly the Decalepis hamiltoni, where the root as well as the flower leaf part is most important. So you can see the high content of the alkaloids and glycosides are used, and this is also used for the medicines and pesticides. And why this plant is becoming endangered? Because of the habitat loss as well as for the over exploitation. So these are all the causes due to that the plants are becoming the endangered or the rare. And this is also one more very important plant, which is the Cocinium fenestratum, which belongs to the family Menispermaceae. So this is also known as the tree turmeric. So why it is known as the tree turmeric? Because the plant stem, what the stems are there. So if you take the powder of the stem, it is look like the yellow color turmeric. That's why the name has given the tree turmeric. But it is a creeper, and it is mainly contain the berberine. So that is why very high content of the alkaloid berberine is there. So that's why it is having a lot of activity. So you can see why this plant is becoming rare, because it is having the low seed germination and the rare reproduction. So if you see the seed of this Coccinum fenestratum, it is very hard, and they are getting the reproduction after 12 years or 13 years alternate. So that's why the plant is not at all growing properly in the general climate, but it is very much available in the Tamil Nadu, Coimbatore, as well as in our Mangalore in Karnataka. Then you can see here, this is the Ilex fasciana. So this is the plant which is rare plant for the Shillong and the northeast area. The all part of the plant, it is containing the high content of the alkaloids, and it is useful for the medicine. So due to this plant having the low rate of the reproduction, it is now becoming the endangered species. So whenever you are selecting any kind of the plant, so definitely you have to go for the toxicity study, because you have to see whether how much dose it is going for giving the safety. So after that, you have to go for the different type of the activity. So this is also one more plant where the plant is leaf is most important, and this is also available in the forest of the Karnataka, Kerala, and Tamil Nadu. And this is now becoming the rare endangered plant because of the overexploitation, habitat destruction, and the low reproduction stage. So this is the things, and this is also you can see here. This is most known plant, Myristica magnifica. So it is belongs to family Myrtaceae. So it is nothing but the known as the tree. It is Western Ghat region. It is widely available, but nowadays it is becoming the rare plant because this root and this fruit and the flower as well as the leaf is most important for the medicinal purpose. So due to this overexploitation and habitat destruction, it is getting the rare or endangered. So this is also one more plant which is most important in the Eastern Ghat region. The leaf is most important for the therapeutic activity. This is also the same problem. So that's why you are having the endangered plant species. And IUCN, they have given the, they have listed the plant as a red label. So these are all plant. Now you can see here this plant, nothing but the Belosynapsis vivipara. So it is known as the spiderwort. So this is spiderwort is nothing but it is having the leaf is 3-4 dimension. You can see here. One is small, one is big, one is this side and that side. So this plant, it is mostly available in case in the Shimoga district, Karnataka. So I have selected this plant for my study for the in vitro culture from the university. I got around 9 lakhs project from this plant. So this is also very important things, and you can do so many activity for this. Nothing is there for this plant. So this is also one more very important plant. It is every known person. You can see the plant. It is Podophyllum hexandrum, belongs to family Barbadeaceae, and this plant is very rare in the northeast region, and due to the overexploitation and habitat destruction. But this plant is available in the Uttarakhand as well as north side and all. So you can Himachal Pradesh and all. So, but nowadays it is going to rare and endangered plants. So this is one more thing, Taxus Ulchania. It is a family Taxaceae. So you can see the Taxus plant is most important for the anti-cancer activity and mainly the ovarian cancer. And due to the overexploitation and habitat destruction, it is now becoming the rare. So this is the plant, the rare plant I have studied in our lab, and we have done the in vitro culture for this all the plants, and we got some results. So you can see here we have taken some media, and this woody plant media we have taken, and we are getting the callus growth as well as the plants growth in the Taxus Ulchania, and you can see the different type of you can see the organogenesis of the plant in the media, where the leaves are coming out in the media from the stem part. And by the SPLC study, you have seen we have isolated the compound paclitaxel, and we have estimated the compound from the plant sources, and isolated plant is giving the higher yield than the plant extracted from the cultivated one or the forest. So this is one advantage for the in vitro culture. It is giving little bit higher active yield than the forest drone, and we are getting the good amount of the pure compound than the others. So this is one. you can see the structure of the paclitaxel. So where the NMR study and mass spectroscopy study has carried out, and this is mainly, it is useful for the anti-neoplastic agent, and it is useful for the ovarian cancer, breast cancer, and lung cancer. So these are all the activity, very important activity that it is doing, and it is this compound has been isolated. Then from this Coccineum fenestratum, it is also isolated the berberine and other compound. And you can see here we have studied the zinc sulfate, and we are supplemented with the IPA, indobutyric acid, and the kinetin in the combination. We are getting the organogenesis, and SPLC study we have studied, and we are using the different type of extract, methanol extract and aqueous extract, and we are getting the percentage of the berberine content in the plant. So you can see the structure. It is also confirmed with the NMR ion mass spectroscopy, where this berberine, it is useful for the treatment of the diabetes, obesity, tumor, and anti-inflammatory activity, as well as powerful antioxidant also. Then we are having the podophyllum hexandrum. So you can see here, this is the plant. It is very difficult to grow in vitro culture. We have done a lot of activity and all. Then we have studied, we have standardized the process, and you can see the growth of the callus from the shoot. It has happened after 28 days of the Na and the DAP in the media. So this is the structure. It is isolated the compound podophyllitoxin, and it is confirmed with the NMR IR and mass spectroscopy, and also it is useful for the potent anti-cancer activity. Then recently in the 2019, this plant has taken, and Baculitis nervosa. So this is the challenging job. It was done by ourselves and our team, where we are using the sodium silicate and the calcium silicate we are using in the media, along with the coconut water and aloe vera gel, so that we can get the proper growth of the root as well as from the shoot. You can see the callus in how the callus is forming in the media, and from this we have isolated the compound, and right now we have isolated the one DNA from this. So this is isolated the DNA, and now the characterization of this DNA is going on, and this is mainly useful for the antioxidant, anti-diabetic, antifungal scent and all. So these are all the activities we have done in our lab. Now this additive technology, so how the additive technology, it is giving the helpful for our herbal technology. So in this herbal field, nothing but the in vitro culture, whatever we are used the glass vessels, that glass vessels we have replaced with these 3D vessels. So this 3D vessels is most important, and this is biodegradable, and how much time you require, you can go for the use. So this is the 3D printing process, is building of an objective in a layer by layer, and it is the fundamental additive rather than the subtractive in the nature. And it is the construction of the three-dimensional object from the CAD model or the digital 3D model. And it is the transformative approach to the industrial production that enable the creation of the lighter, stronger, as well as the biodegradable. So this is the technology nowadays we are using in the in vitro culture technology, so that it is very much helpful. And this technology, it is also important for the pharmacological aspect, where we are using some sort of the artificial organ, so that we can reduce the animal experimentation, so that it will be helpful for doing the experimentation by these techniques. So this is the 3D printing techniques we are using in the plant tissue culture. So this method is tested physically to hold the plant upright, thereby facilitating the liquid based routing. So if you are using any kind of the liquid media, so if you are using the plant, so plant will be declined. So what will happen? We are keeping the plant straight. So for that, we are making some sort of the holes in the vessels, and we are keeping the plant in a straight position, so that plant can grow normally. So this system was developed with the Malus domestica, then Betula lenta, as well as Musa species, nothing but the banana, using the static liquid culture as well as a rocket-based temporary immersion system. So we are, by these techniques, it is very helpful for the rooting of the plant, as well as it is more comfortable from the spreading of the rooting, as well as it is reducing the labor requirement, and also the preventing the root damage. So that's why the plant will grow very fast, and the development of the bioactive compound in the plant, it will be very easily you can identify, as well as you can go for the isolate. So this is the technique. You can see the biodegradable 3D printing vessels. So in these culture vessels, you can see here we are using the this is the liquid media we're using, and in these small holes, all the plants we are keeping, whether the plant getting the lot of space for the rooting and all. And this is the you can see here the culture vessel, the root stand, and two pieces. The root stand is there. You can see different types are there. Root stand is there, having the dimension also 73.3 mm, and the length is around 225 to 0.3 mm. So this is the length we are using for the culture vessels. So this is most important for nowadays as innovative in a plant tissue culture. And you can see here, this is the growth of the plant. This is experiment already done. It is for the cherry which, then this is the B, which is based on the apple, and C is the banana tree. When you can see the growth of the banana and the space in between the plant, they're getting a lot of space, so that rooting is very easy, and the growth of the plant also very fast. So this is the 3D printing technology. Here we can go for the establishing the LED light in the individual culture vessel, so that plant can grow individually. They can grow very nice, very nicely, rather than the how we are doing the conventional method plant tissue culture. So this is the techniques you can follow for the next generation. You can get a lot of demand for this kind of activity, and you can isolate the bioactive compound from this kind of the endangered plant species by growing plant tissue culture. And we are getting a lot of demand for this, and we can go for various kind of the high impact factor publication. So you can see here the culture vessel, the LED light, we are having fluorescence light. So different type of light we are using, and for the plant growth. And whenever the plant is plant growth will be over, you can reuse these vessels once again. There will be no chance for the breaking. So whenever you want, you can go for reuse. So there is a less labor cost, and as well as you can use the longer time. So you can see here the tobacco artemisia plant. It is cultured in vitro condition, with having the leaves with the various kind of the light spectra we have done. So this is, it is taken from the reference. It is published in 2017 paper. And you can see for the 3D printing technology in the Bangalore, there is a company known as Arium 3D, then Altium Technologies, then Ansu 3D Technology Private Limited. So these are all the technology companies. They are having the 3D printing technology, and they can giving the chance for doing the so many activities and all. Even in Telangana, there is so many companies for the 3D printing, where the Altium Technologies Private Limited, then Marker Global, then Garuda 3D, then Print My CAD. So these are all the companies they are having. So if anyone interested to do the 3D printing technology for your research, so please you can contact to them, and you can get the suggestion how you can go for the activity and all. Then as per now, come to the last slide. For the conclusion, I can say that phytoconstituent mainly it is utilized for the lead compound. Then identification of the plant constituent by the screening method is most important to identify the preliminary lead compound. Then pharmacological screening followed by the clinical trial is important for the development of the lead compound. And for the getting the raw material, as well as for the raw bioactive compound from the isolated plant, we have to conserve the endangered plant species, so that we can use the plant for the future generation. Then for this, we can go for the in vitro alternate plant tissue culture technique, so that you can go for the mass propagation for the endangered plant, and you can go for the isolation and discovery of the lead compound, and you can efficient of the modern 3D printing technology we are using for the in vitro plant culture for the getting more amount of the bioactive compound, and it will be very easy to isolate. So it is, I can say one thing, it is very important on term, you can see here, education took us from the thumb impression to the signature, but technology took us from the signature to the thumb impression. So it is not like that we are the illiterate. We are going by using the different type of the technology, and by using the thumb impression, we are using so many things and we are doing the wonder. So you can acknowledgement, you can give the first my sincere thanks to Dr. Rajiv Daya sir, APP President, for giving me an opportunity to deliver a lecture. My sincere thanks to Kripanjali College of Pharmacy Principal and Management for giving me an opportunity for the laboratory facilities. And my heartfelt thanks to Principal Organizer of St. Paul's College of Pharmacy, Telangana, for giving me active support and giving me opportunity to give my thought in this platform. Thank you.
Sir Kuntaldas sir, very nice presentation.
Thank you, sir.
So you given a very elaborately about the topic, and as you see in the nowadays, the total people are going back towards the natural sources.
Yes, sir.
And you highlighted what are the different active constituents that is present in the natural sources. And you explained all the different things like what are the different types of the rare plants which we can use it for the research work for the budding students, budding research students.
Yes.
And I am having only one simple question, sir. Whatever you are doing the in vitro technology or 3D printing technology,
Yeah.
after growing that technology, whatever the active chemical constituents is there, that is similar to that of the natural things or any lack?
No. It is similar to natural things only.
Okay, so because as you've seen in the case of the chickens and all these things also, we will have two different varieties. Those the things which are grown in the outside may not be having that much values as compared to that of the natural one.
Yeah. Actually, sir, what we are doing here, we are using only the vessels. So instead of the glass apparatus, we are using the biodegradable 3D printing vessels, so that we can use for the any kind of the liquid culture media, so that plant can grow easily.
Okay, sir. Thank you. And any other questions from participants?
Sir, I have a small question, sir.
Yes, sir.
Sir, your lecture is really an eye-opener. I'm really happy to be part of it, sir. Sir, you have given some species are really endangered.
Yes,
it's very sad to listen to that. Can you suggest some measures how to restore those endangered wonder herbals, sir?
Sir, the thing is, first of all, we have to conserve the pollen grains as a cryopreservation. You can go for the seed or can go for the pollens. And there is a research center. It is there in the Bangalore in Indian Institute of Horticultural Research. So they are having the different type of the pollen culture. They are having the seed culture, and they are having the cryopreservation. So any kind of things you can go for preserve, and you can reuse whenever you want.
Yes. Thank you, sir. Thank you. Sir.
Yeah. Thank you, Professor Das, for the nice lecture. I have one question. So can you enlighten us on herbs used in the present COVID situation?
COVID situation, you can use the Tinospora cardifolia, which is known as the Amrutavalli. That is most important. Then Glycyrrhiza glabra, nothing but the glycyrrhizin. Then we are using the people leaf, nothing but the black pepper tree. So these are all the very important, along with the ginger and all. So it is as per the Ayush, they have recommended this 5-6 plants, which is you are using for the preliminary precaution for this COVID situation.
So whether these are they are using alone or in combination?
No, in combination.
Okay, combination.
Yes.
Okay. Thank you.
Thank you, sir.
Sir, Kuntaldas sir, it's very happy to be with you today. So it's really very enthusiastic session, sir. It's my interest, like area of interest also, these herbal drugs and all. Especially the way you projected the awareness about additive technology, especially additive 3D technology and the utensils, biodegradable things and all. Really, it's a very new thing for many people. So it's an awareness session, sir. Purpose has been served fully, 200%, I can say. And regarding this endangered species, sir, so Raju, sir, even I want to discuss this point. So like uniquely, many aroma centers in Hyderabad, also Center for Aromatic Drugs is there. So why can't we from APP, we can start initiative to preserve these things, like to give awareness to the public about these endangered natural species. And we can, our newsletters and all, we can cover this part also, so that people will aware of it. At least where is the source of that? Like sir said, some manure, some center is there.
Yes, that is the main problem. We are not getting the raw material source for doing any kind of research.
Yes, sir. Yes, sir. Because I believe that India is the heritage of a lot of these kind of plants, sir.
Yes,
If we really explore,
yes,
sir, with a systematic manner, with the help of experts like you, if we take the lead, definitely we can explore a lot of things to the future generations. I hope.
100%, madam. Any help, anytime, please tell me.
Thank you, sir. Thank you, Rajiv Dahiya, sir. And so please think about my proposal, sir.
Yes, sir.
Yeah, thank you, madam. It is a good initiative. So actually, our journal and our magazine. So I'm also lacking some persons who want to work with me. So it is I welcome anybody who is interested and if want to elaborate on any of the health related activities. So I'm ready to include them and at editorial level. So in the board, and you are free to. So any idea is welcome which can be beneficial in health sectors, and as pharmacists we can play the role in the health sciences. So thank you.
Thank you, madam.
Sure, sir. Sure, sir.
Thank you, madam.
Thank you so much, sir. It's indeed a wonderful session. Thank you so much for your time.
Thank you, madam.
Now I'm privileged to introduce our next speaker, Dr. S. Satish Kumar, sir. Dr. S. Satish Kumar, sir, is presently working as a professor and HOD, School of Pharmaceutical Sciences, Department of Pharmaceutics, Wales Institute of Science and Technology and Advanced Sciences, Chennai, India. Sir has done PhD in 2009 from Kakatiya University, Warangal, M.Pharmacy in Pharmaceutics from Dr. MGR Medical University, Chennai. Sir has 75 publications in national and international journals with H-index of 7 and ITIN 5. Sir has attended more than 40 conferences and 17 training programs and published 1 book. Sir has guided more than 10 PhD candidates, 30 postgraduate students, and more than 40 undergraduate students. Sir's area of research is the design and development of nano-drug delivery systems for targeting brain. Sir has awarded with Honorary Doctor of Letters from University of South America, Pennsylvania. With this brief introduction, I now invite and I request Dr. Satish Kumar, sir, to please start the session. Over to you, sir.
Good morning to all. First, let
morning, sir.
Let me share my screen. Please
Yes.
enable screen sharing, madam.
It's not pinned. Now it is pinned. हाँ सर मैं होस्ट करती हूँ सर
क्लीन शेयर फर्स्ट
Now sir is host. You can share the screen, sir.
Yes. Is it visible, madam?
Yes, sir, it's visible, sir.
Yeah, good morning and welcome to my small lecture session. This lecture session is tailor-made for young researchers who can understand and want to make some fruitful output out of their research, starting from undergraduate to PhD program. So that the title I had chosen is Nanoparticle, a successful tool for delivery of antimicrobials across blood-brain barrier. So in the title, I had used the word successful tool for delivery. Delivery of drugs. Drug delivery systems are existing very long back, and we are using different types of drug delivery systems. And what is a successful drug delivery system? If you put that question in this title itself, I can say that a drug delivery system is successful if it can do two things. One is it should deliver the drug at the right site of action where it is required. And secondly, it should give a consistent concentration of drug required for producing the therapeutic action or exerting the therapeutic action. If a drug delivery system can do these two things, then we can call that drug delivery system as a successful drug delivery system. So in this presentation, I am showing you that these nanoparticles as a successful drug delivery system for delivery of antimicrobials across blood-brain barrier. Say, again, one more thing I want to explain with this title itself is why this antimicrobials and blood-brain barrier, these two things are taken for our discussion. One is the blood-brain barrier is an important barrier, natural barrier, which protects the brain from external impacts, not only by the contents foreign bodies coming into our body, but also from those internal chemicals and other metabolic products which are running in our circulatory system. They do not reach the brain. So it's a natural barrier. And even these internal components are not even sent to the brain by this blood-brain barrier. How this we can expect that these antimicrobials will enter into the brain successfully. So we have to treat certain infections, particularly like infections like meningitis, which are occurring in brain. We have to put a lot of effort to transport this drug across antimicrobials across this blood-brain barrier. So this discussion will be oriented towards application of nanoparticles for treating infections in brain. So moving to the next slide. First, I like to say a few things about the brain. The brain is kept very safely in a cranial system or cranial cavity where the external impacts do not reach. Also, it is protected by an barrier which is rich in membranes, which will not allow the external materials, foreign bodies, and even some of the internal components which are circulating our normal circulation will not reach the brain. So the brain, it looks like a pinkish gray tissue containing a lot of neural networks called neurons, that we call it as nerve cells. The cortex is involved with the memory, recall, reasoning, and personality. You know that different parts of the brain, you take forebrain, hindbrain, or cerebellum, thalamus, whichever a part, it has its own function. So this presentation will not be sufficient enough to discuss the functions of brain, different parts of the brain that take over in our life. But for example, I had shown one or two functions that brain is carrying out. Like that, the hippocampus will process new informations and register in your brain. So each and every part of the brain has its own functions. So basically, it is a very important organ in our body. Next is blood-brain barrier. I want to say a few things about this blood-brain barrier. Like any organ in our body, our brain also receives a lot of blood. It is having a rich supply of blood capillaries. Blood vessels are running across the brain. But this system, network system, is having a special protection. The contents of the blood in the blood vessels or blood capillaries does not exchange with the brain as happens with the other organs. Every organ has an unique property. For example, the kidney, the function is particularly purification of the blood. Likewise, in the lungs, it is just oxygenation of the blood. Likewise, every organ has its own function. Accordingly, the blood vessels are also designed there. But in case of brain, even though there is a rich supply of blood vessels, these blood capillaries will not exchange all the contents of the blood into the brain. It will selectively send the required materials alone into the brain, and the remaining will stay inside the blood capillary itself. So only required nutrients, then certain minerals, selective drugs, etc., only can pass through the blood-brain barrier. This blood-brain barrier is nothing but the blood vessels are surrounded by extension or modification of the glial cells that is called as astrocyte. So the thick junctions formed by these astrocytes, pericytes, and endothelial cells together form this blood-brain barrier. So blood-brain barrier is nothing but a tight junction around this blood capillaries, which are made up of the extension of astrocyte, extension of glial cells, what we call it as astrocytes. In addition to its pericyte and endothelial cells together form a tight junction. It will not allow all the materials to pass into the brain, which are in the blood. So this blood-brain barrier is a major restriction for pumping the drugs or transporting the drugs from the blood to the brain. So whatever we inject or administer, it will be available in the normal circulatory system, but it will not reach the brain. Only selectively few drugs. There are few lipophilic drugs that can easily pass through this blood-brain barrier and reach the brain. Most other drugs, even though if you give in higher concentration also, it will remain in the blood, in the central circulatory system, but it will not be available for the brain. So this is a part of the natural protective system or protective protocol designed by the nature. So in case of certain infections occurring in the brain, for example, for this study, for this discussion, we can take meningitis. And if this drug required for treating meningitis is given into the body, either by oral or by injection, it will be available in the circulatory system, but it will not be transported across the BBB and reach the brain the required concentration. Okay, now we'll move on to the next slide and discuss more about this meningitis and again come back to the brain. So meningitis, what is meningitis? Meningitis is a devastating disease that remains a major public health challenge, which is caused by different microorganisms. We cannot say that a particular microorganism alone is causing this meningitis. This the meninges or the membranes that cover the brain and the spinal cord. And if any inflammation occurring on this part of the meningeal membrane or the liquid surrounding it, then we can call it as meningitis. The most common causes are viral infection and bacterial infections. See, the main thing we can think about this meningitis, or we want to discuss about this meningitis, is it is an infection that is occurring in the meninges or the area surrounding this meninges, that is due to viral or bacterial infections. So bacterial infections are very common when compared with the viral, although viral infections are persisting, that are not so common. Only on selected areas or in a selected season, only this viral meningitis are occurring. So it is comparatively having a less proportion. But very commonly seen is what we call it as the bacterial meningitis. What this bacterial meningitis will do means it causes infection around the meninges and it affects the brain, causes the infection in the brain, sepsis in the brain, and the neurons will be dying. And if we are treating the meningitis very seriously with certain drugs, sometimes the patient is recovered. In most cases, we could able to recover the patients, but the damage caused in the brain by the sepsis or the infection will make the patient a little bit socially, mentally challenged because of the damage caused by this sepsis. So commonly, bacteria like Streptococcus pneumonia, Neisseria meningitidis, Haemophilus influenza, Listeria monocytogenes, and group B Streptococcus, and even Escherichia coli, E. coli, can also cause the infection in the meningococcal area. So in this slide, I had shown how a normal brain and how an sepsis brain or meningitis brain will appear. See, I already told it is a pinkish gray organ. Normally in a healthy brain will be in pinkish gray color, which is shown at the left-hand side half of the screen. And in the right-hand side half, you can see a greenish tint over the brain, which is occurring due to the sepsis of meningital infection. So if you see the autopsy of a patient who died out of this meningitis, we can see a greenish tint. See in that photograph, the membranes are slightly moved and they are showing the brain, surface of the brain. Instead of its pinkish gray, we can see green tints here and there surrounding it. So this type of infection leads to formation of sepsis around the brain, which will collapse the total functioning of the brain step by step. Some more information about the meningitis. It can be caused by different types of pathogen, including virus, fungi, and even bacteria. Together with sepsis, meningitis is estimated to cause more deaths in children. It's very important information. We can understand that children below 5 years are widely affected by this meningitis, and the death rate is also very high in the children. The survivors suffers equal with considerable social and economic cost, because due to the damage caused by the infection in the brain, the patients, even after recovery from meningitis, they will be having some mental illness that is due to the neuronal degenerations occurring due to the sepsis, which will make the patient fully not recoverable, or even it will take very long time to recover to the normal stage. Recognizing the global importance of this problem, WHO and its partner countries had designed a strategical vision to eradicate or defeat this meningitis by the year 2030. Tedros Adhanom had recently told about this in 2021. He talked to the media about this meningitis infection prevailing in various countries and its deadly sequences. So the countries had, the WHO countries had also have a vision to stop this, defeat this meningitis by 2030. The studies from India says that meningitis is also one among the major reasons for death of children below 5 years. So this is for our information. We have our children in our home, or even our friends and relatives who have children. If meningitis occurs, how we can identify what are the symptoms? Because infection in brain, infection in any other organ is different. Infection in brain is different. So what are the symptoms that we can find? Fever. Fever is normal in any infection, but here there is a specific symptom. The hands and feet will be cold enough, and the remaining, if you test the patient, they will be having a fever. So normally fever means you will not feel the chillness, you will feel the only a hot surface in the body. The body temperature might have been elevated. But in this particular meningitis patients, we can see fever If you measure using a temperature, internally it will indicate the fever. And the hands, if you touch the hands of the patients, they will feel very chill. Likewise, on the feet also. Then the children will be feeling not being being handled. They don't want to handle by the parents. They don't want to go to anybody else. They will be try to separate from them. Then rapid breathing and grunting issue, shortness of the breath, and fastly they will breathe. So that you can also find this is, which is normally not a symptom in normal fever at all. Then unusual crying and moaning. Okay, children they will be crying for everything, but still unusual crying and moaning can be considered as one of the symptom. Then stiffness in the neck and dislike bright light. See, it's a disliking of bright light is a normal symptom for any neurodegeneration, for Alzheimer's disease or even Parkinson's disease, Huntington's disease, any neuro disease disorder you take, they won't like a bright light. But this disease, when you can combine these other symptoms like fever with chillness in hands and legs, and rapid breathing, unusual crying, along with dislike to bright lights, with stiffness in the neck. The children cannot turn the neck here and there, left and right very quickly. They will feel some stiffness and pain in the neck. And refusing food and vomiting. Refusing the food. Children will be not liking to have the food, and sometimes they also show vomiting. Then drowsiness, floppy, and unresponsive. If the child is unresponsive, say normally fever will be there, and if the child is responsive, if you call the child, it's answering, it's active, means that fever does not cause anything. But if the child is unresponsive, we should have a great attention to that. Not only this meningitis fever, for any fever that cause very drowsiness in the body, and if the patient is unresponsive to the instructions, then we should consider that fever as a very important one. And the next one is pale, blotchy skin with spots and rashes in the skin. See, this is a very important symptom, which is different from normal fever. This type of symptom is not seen in other fevers, normal fevers or normal infections. There will be round spots, rashes on the skin. If you put a glass on that glass tumbler or that skin and press it, you can see that the redness will spread. This type of rashes are there in the skin is very peculiar and very specific with this meningitis fever or meningitis infection. But one thing I have to remember, this rashes will not appear as soon as the infection is occurred. It will take some time to, or we can say that after 1 week or 4 or 5 days only, this type of symptoms will be visible. So you can understand that the infection had got occurred a few days ago itself. That's an indication for that. And tense and bulging fontanelle. That means there will be a bulging in the head. If you find unusual bulging in the head on the fontanelle portion of the brain, that head, we can understand that something is wrong in some infection had taken place in the brain. Then convulsions or seizures. These are all together put. See, it's not one symptom. There are various symptoms. All the symptoms may not be visible to every patient. Combination of these symptoms have to be taken into account, and we have to understand that, or you can suspect the presence of meningitis. So why I'm saying this much about the symptoms means this meningitis is having the risk of death of children below 5 years. Even at a higher age, the patient will say that he's having pain, he is having discomfort in the head. So all those things they can able to communicate properly. But the children, they do not have that specific mind of communicating these things to the parents perfectly. So we have to analyze when fever occurs. We have to analyze all these symptoms, because the death rate is very high in this children below 5 years who had been affected with meningitis. So I took all this together for you. And this will be a helpful thing, not only for your research, but also in your personal life to help others to identify meningitis. What difficulties are encountered in treatment of brain infection? Let it be meningitis or any brain infection. What sort of troubles are there for us? Say, brain, spinal cord, and surrounding are being infected by large spectrum of germs or microorganisms. It is not only for meningitis, for many other microorganisms may also attack these parts. Treatment of infection in brain is a major challenge because of the it's a unique type of organ. The restriction of the drugs to enter into the brain by BBB, and only lipophilic drugs are more freely entering into the brain, whereas most other antimicrobials are falling under the category of hydrophilic in nature. And hence, there is a need for application of novel drug delivery system like nanoparticles to enhance the pumping of the drug into the brain. So what are the strategies we have to use, or we are presently having, to pump the drug into the brain? So I told that BBB is there. It restricts the flow of drug into the brain. And what other or what strategies are available for putting the drug into the brain? First one I want to say is the chemical drug delivery system. That means I told that hydrophilic drugs are not easily permeable across the BBB. So we can convert by means of a chemical modification by attaching a lipid mediated group or a lipid functional group onto the chemical moiety of the drug, and you can enhance the permeation. And another one is the biological delivery systems. We can there are pre-engineered drug delivery systems like resealed erythrocytes and all. So we can use some biological drug delivery systems or endogenous transporters can be used for putting the drug inside and pumping into the brain. Then the third strategy is disrupting the blood-brain barrier. It's very dangerous. Of course, in some cases, if there is no other go, we have to follow this strategy also. That is directly putting the needle across the BBB and putting the injection of the drug into the brain, or by using a saline solution of a particular concentration, the BBB will be disrupted temporarily. See, it's very dangerous because when there is an physical disruption of blood-brain barrier, that does not allow the drug alone. It will be open for all materials. All metabolites in your blood will be transported across this BBB and entering into the brain. And any chemicals, any other drugs that are available in the circulation will be also taken into the brain. So in addition to that, if any other infections, that is microorganisms are circulating in our blood, they may also enter into the brain and cause infection. So disrupting the BBB physically is not that much accepted, unless and otherwise it is very essential, and it is the last step that we have to take. And receptor mediated delivery. A lot of receptors are available in this BBB, and we can able to transport the drug across the BBB using these receptors. By utilizing these receptors, we can transport the drug across the blood-brain barrier. I'll show on the next slide the diagrammatic manner. So we can see across the membrane that of the cells plus endothelial cells, we can transport the proteins like glucose, amino acids, ketones, etc. Across the membrane itself, because these membranes are permeable to certain things. And the second one is diffusion. Diffusion can also take place across this membrane, depending upon the concentration gradient. And the third one, we can see there are intercellular junctions. There, the junction gaps are there, means these endothelial across these endothelial cells, it can be pushed into the brain across these fine tight junctions and pump the drug into the BBB. And receptor mediated, as earlier we discussed, you can see here the receptors available in the endothelial cells can be utilized for transporting certain drugs, transferrin, insulin, then conjugated nanoparticles, etc., can be transported across this endothelial cells by using the receptors. Then adsorption mediated. We can also transport certain nanoparticles, particularly the surface modified nanoparticles, by attaching or certain drugs onto the surface, and then you can push it into the brain by means of adsorption onto the surface of the nanoparticles. So next, I want to move on to these nanoparticles, how we can successfully use these nanoparticles onto the delivery of drugs across BBB. Say, most of you may know about very well about these nanoparticles, and I don't think that very heavy description is required for that. Anything below 1000 nanometer we can consider as nanoparticles, and they may be amorphous or crystalline in nature. And it's a very versatile drug delivery system. Particularly, polymeric drug delivery system nanoparticles are very successful because of their good stability, and both internally as well as physically, when they are stored also, they will exhibit good stability. So I'm not going to explain much about these nanoparticles, the basic definitions and all those. It's a very fine particle. For undergraduate students, purpose of the undergraduate students, I had made a comparison with the hair. If you take a cross section of the hair, human hair, it is around 60 micrometer. In that one hair, cross section of the hair, we can have 1 million particles of nanoparticles, 1 million nanoparticles with 600 nanometer of diameter, and 1 billion nanoparticles of 60 nanometer diameter. So you can see how fine these particles are. What this comparison says to you is how fine these nanoparticles are when compared to hair. One cross section of one hair can have 1 billion of nanoparticles, which are 60 nanometer diameter in size. So these could able to do a lot of miracles, and they are being lively shown in different experiments too. Again, why nanoparticles we are specifically using for brain? So many other drug delivery systems, liposomes are there, and microparticles are there. Why we are using this, or we are preferring to have nanoparticles for brain? Basically, smaller in size, of course, stable. The stability not only when after a biological application, even when during its storage also, we could able to find a very good stability, particularly polymeric nanoparticles. Then they are very safe. In the previous talk also, Dr. Kuntal Das professor had told how this additive should be biodegradable. Likewise, this nanoparticles, which are made up of certain biodegradable polymers, are very safe, and they do not cause any toxicity, in fact, and they are completely disposed after the use on the body. Then they are very precise in their action. We could able to make some modification on the surface, and we can make it can convert it into active nanoparticle, and we can make active drug targeting very precisely to the site of action. So smaller, stable, safe, precise, and active are the basic reasons for selecting this nanoparticles for brain. Is there any other advantages? Yes, it will be having a very good drug water solubility, apparent ratio of drug water solubility is high, so that we could able to entrap more amount of drug into the nanoparticles. Likewise, the site of absorption and the enhancement of the mean residence time at the particular site of absorption will be very high, so that the concentration gradient inhibition will not be available for this nanoparticles. It will be easily moving away after absorption, and permeation across the cells are very high. Then we can also control, this is a very important point, control of the release kinetics is also possible by means of nanoparticles, because polymeric coats are there. By hardening or increasing the thickness of the coat, we could able to control the release kinetics of the drug from these nanoparticles. It's a very important property that we'll usually manipulate upon to achieve the perfect target as well as concentration. And achieve targeted drug delivery system, as already told, it is having a very good targeted drug delivery system too. Very specifically, we can target to a site or a receptor or a cell, or even a cell organelle can be targeted by using a nanoparticle. Role of nano drug delivery system in transport of drugs across the brain. It's having multifunctionalization. That is, we can convert a nanoparticle into a very active nanoparticle, which will deliver the drug only in the particular site of action, so that this cannot be done by most other novel drug delivery systems, so that the drug will be released on a particular area alone, particular cells or receptors alone. So this is an added advantage of nano drug delivery system. As repeatedly I told, it's having a very good chemical, physical and biological stability. Then feasibility of incorporating both hydrophilic and hydrophobic compounds or drug materials into the nanoparticles. It can be administered by various routes, not only by injection. It can also be used for oral administration, inhalation. That means nasal drug delivery system is also possible by means of nanoparticles, in addition to the injections. So we can improve the functionalization by adding ligands. That means not only polymeric ligands, but also you can use proteins and other materials, biologically active materials, to enhance the efficiency of targeting. Now we'll take one sample drug, ansamycin, as a model drug for incorporating into these nanoparticles. This ansamycin is also used for treatment of meningitis. So I selected this drug. It's having a, the important thing I want to focus upon here is its biodistribution. Normally this ansamycin, when it is given as a drug injection, about 85% remains in the plasma and only 4% reaches the brain. We had optimized prepared a nanoparticle made up of PLGA with this ansamycin in a 10:1 ratio of aqueous phase to organic phase. It's an optimized formula. We had worked a lot to get this formula optimized. After a few experiments, we could able to achieve this. So in this PLGA nanoparticles, we had loaded ansamycin, and the particle size was around 125 nanometers with minus 25 millivolt as its zeta potential. The drug release kinetics were carried out. See, the pure drug in phosphate buffer as well as plasma was being released very quickly. 100% of the release was there, and it has happened within 1 hour. But in case of this nanoparticles, we could get a continuous release for up to 36 hours in the phosphate buffer saline as well as plasma. We had also carried out the pharmacokinetic studies of these nanoparticles loaded with ansamycin. The nano formulation was injected into the tail vein of the mice at 25 mg per kg. Blood samples were taken at predetermined time and analyzed for the drug content by using HPLC. And we had used a software called Vinolin version 3 for studying the computing the pharmacokinetic parameters. The plasma concentration versus time profile was made for both pure ansamycin drug and for the nano formulation of the ansamycin drug. So you can see here up to 24 hours, we could able to find this traces of ansamycin when it is loaded in nanoparticles, but whereas the normal ansamycin drug had been eliminated within 8 hours of time. Next is the tissue distribution studies. We had injected a particular amount of the ansamycin drug in one group and ansamycin loaded nanoparticles in another group, and we had extracted the organs, heart, liver, lung, kidney, and brain and analyze the amount of the drug by HPLC after extraction. See, the pure enzymycin drug is indicated in the orange color, where in heart there is a little bit increase in penetration, but whereas in liver you can see there is a drastic fall in the amount of the drug distributed. You can again see in brain it has increased multiple times. From 4%, it might be extended up to 60 times approximately. To a very high level, there was an increase in the drug loaded nanoparticles could able to push the drug into the brain. Likewise, we had carried out various enzyme studies. The enzymes are affected when the meningitis are induced, and after giving the drug, we could able to retrieve the normal activity of the enzymes, and nanoparticles had very well established to the close to the normal level, and that indicates that activity also had increased. Not only the concentration, but also the activity could also be increased by loading the loading the enzymycin in nanoparticles. Again, confirm, we have to go for pathological study by staining the brain histopathological slices. We had seen there is a lot of recovery of normal like recovery of the damaged brain very quickly when compared with that of the pure enzymycin drug. So what are the major outcomes of the study? That's what I want to highlight here. What I want to say in the nutshell is there was an increase in the Cmax of the enzymycin in the blood, as well as the uptake of the drug in the brain was very high. The brain tissues were loaded with more amount of, say, in brain around 4,496 nanograms of drugs been established after loading into these nanoparticles. It's a very high increment. A significant increase in the bacterial load is noted after 8 hours and stayed very high for 36 hours in positive control. The same reversed it using nanoparticle treated animals. The altered body temperature, heart rate, and blood pressure are also reversed. That indicates the activity. Likewise, the liver function also has been increased, because the amount of the normally you can see in the previous slides. Here you can see the liver, the pure drug, the concentration is very high. The concentration is very high in the liver, and it is reduced to nearly half after loading into nanoparticles. So this indicates that the liver also gets the benefits of loading the enzymycin in nanoparticular formulation. And these are the publications which are made out of this work, and the patent is pending the patent office for approval. And we had not started this work of loading the nanoparticle the antibacterials or antimicrobials in nanoparticles. It was started even at the stage of 2004 itself. We published a work, formulation and physicochemical evaluation of polystyrene nanoparticles containing cefotaxime sodium, again, which is an antimicrobial. So I hope that in conclusion, we can understand there is an enormous potential for loading this antimicrobials in nanoparticles. See, it is much better than drug discovery to load these drugs in nanoparticles and enhancing the efficiency. So acknowledgements, I'm very much thankful for Dr. Rajiv Dahiya Garu for giving the opportunity. Dr. Sunita Dahiya is the General Secretary of this APP, and Dr. Hemendra Gautam, who is President of Association of Pharmacy Professionals. And also I wish to thank APP Karnataka Spread Branch, APP American Branch, International Branch, and APP Analytical Chemistry Division for providing this opportunity. And I thank Professor Kiranmai Mantava, the convener of this conference, and the management, organizers, faculty members, and students for providing this opportunity. Once again, I thank for this opportunity for APP and St. Paul's College of Pharmacy, and thankful for all the listeners for patient listening. Thank you very much.
So Professor Satish Kumar sir, your presentation was very nice.
Thank you.
And you elaboratedly explained all the things like nanoparticles individually and the blood-brain barrier individually. And also you told about the drug delivery system, how these nanoparticles are useful for crossing the blood-brain barrier. And also taken an example of enzamycin. And one simple question I want to ask to you, any hurdles you observed in preparing the nanoparticles of enzamycin?
Sir?
Any hurdles you observed in preparing nanoparticles of enzamycin in your laboratory?
Yes, of course. A lot of yes, sir. A lot of problems we had experienced. I had told only PLGA nanoparticles here, but we tried with more than 7 polymers. Among that, 3 polymers had not yielded any successful nanoparticle because of their pre-formulation problems, because they had certain interactions with the drug, and even the functional group of the drug was changed with a particular polymer, so that we could not able to achieve successfully. So after repeated experiments, we could able to get this PLGA nanoparticles, which could able to be successfully loaded, and we have to modify a lot of things with the procedure also. Normally adopted procedure was not fit for preparing this for using for these antibacterials. We could able to modify after a lot of experiments. We changed the certain things. The poloxamer concentration was changed, and then the amount of organic solutions used were changed, and finally we could able to get this. So it was a very big trouble for us. Again, carrying out animal studies is also not easy. Pharmacokinetic studies, we could able to do it. It took very long period for doing this animal studies and to complete the pharmacokinetic studies. That was also a very difficult situation during the Corona period and all. We got a really hard situation to get the animal studies done.
So whatever your efforts will not be wasted, and hope that you will get the patent in a short span of time. As you mentioned that it is in the pending stage, and wishing you all the best. And whatever your topic chosen was very nice for the theme of the 17th Indo-US Conference, recent advances, global trends in pharmaceutical sciences. Wishing you all the best.
Thank you.
Sir, I had a small query, sir.
Yes, sir. Please, sir.
Sir, when you are showing the slide of drug concentrations in various organs of the body, you have achieved a significant concentration in the brain. Of course, the brain is the organ which has been targeted, but the concentrations in the kidney is very low. So how can we relate this one to its elimination profile, sir?
Elimination profile, actually, there may be a definite change in the elimination profile also. One more thing, so you very much focused towards the particular drug distribution studies. I want to say one more thing here. You might have noticed that there will be an increase in the distribution of this enzamycin in heart also. That was really worrying for us because usually it will not happen. Because of the property of this nanoparticles, I don't know. We are not very clear about the mechanism involved, but there was we found that there was an increase in the amount of the drug which entered into the heart also. Likewise, definitely there will be a change in the profile of elimination because the distribution pattern has changed. We have to carry out a detailed study on the elimination of elimination profile of the drug which is loaded in nanoparticles separately, sir. Unless and otherwise, we cannot prove the safety of this formulation 100%. And one more thing is there may be variation in the the metabolites also, because when the amount of the drug reaching the liver is less, there may be definitely there will be a change in the amount of the metabolites eliminated from this nanoparticle formulation also. They're expecting that also. So as detailed study is required, unless and otherwise, we cannot directly say anything about the elimination profiles.
Thank you, sir. Thank you very much, sir.
Yeah, Professor Satish, Dr. Daya here. So thank
Yes,
you
sir.
for the educating us on the nanotechnology. So just a general question, what future you look in for this nanotechnology field in the present COVID situation and the medication development? If anything you can
Definitely
focus on.
there is. Yes, sir. Definitely there is a scope for enhancing the potential of existing antivirals. Not only antivirals, but also putting these vaccines in together in the nano formulations definitely will improve the activity and the immunity enhancement will be much more higher than normal vaccines. So vaccines as well as antivirals both can be used in this nanoparticle platform, and we can enhance the activity. That scope I widely feel in this, and we are also planning, if possible, we'll do some antiviral drugs we will test.
So thank you. So
We had selected
hope to see
antiviral drug, and then presently we are working on it.
Thank you, Dr. Satish.
Thank
So
you.
hope to see you some good research on the topic in the future, so that we can also contribute in COVID treatment to some level. Thank you.
Thank you very much, sir. Thank you very much for the opportunity, sir.
So any questions from the participants?
Please feel free to ask the questions, because I may not have out of time, or because due to some reasons I might have skipped some part. If anybody have any question, they can feel free to ask the questions.
Thank you, sir. I think the participants don't have any questions. They have understood it very well. Indeed, it's a very wonderful session, sir, especially for the research scholars who are working under the nanotechnology. It's really a great session, sir. Thank you so much for your time for today's session.
Thank you, madam. Thank you for the opportunity.
Thank you, sir. Next, moving on to the next session, we have now Professor A. Sridevi Madam. I'm honored to introduce Sridevi Madam for today's session. Professor A. Sridevi is an alumni of Kakatiya University, Warangal, Telangana. Madam has 25 years of teaching and research experience, served as head of the Department of Institute of Pharmaceutical Technology, Sri Padmavati Mahila Vishwavidyalaya, Tirupati. Presently serving as in charge head of Physical Science Department. Madam specialized in phytochemical analysis, screening of natural products for various biological activities. Proving her scientific competence, Madam has completed 5 research projects with a research grant of around 70 lakhs. Currently, 1 more project is in pipeline. She has been guiding 8 PhD students and 6 PhD students were awarded. Supervised 45 PG project students, and Madam has published more than 60 research peer-reviewed publications and contributed 4 chapters in various books from reputed publishers. Dr. Sridevi is life member in professional bodies such as IPA, APTI, and APB. As a BOS chairperson, played a crucial role in implementation of PCI approved PG curriculum and introduced MOOCs course in PG program. Madam is a recipient of AICTE CAYT Award, UGC Research Grant, and State Best NSS Program Officer Award. Madam is a member in several prestigious bodies like Member Fact-Finding Committees for Acharya Nagarjuna University, Selection Committee Member for Sri Krishnadevaraya University, IAEC member, Secretary of SPMVV, IAEC member for several institutions, CPCSEA main nominee for Kuppam University, PES Institute, Narayana College, Nellore, and National Assessment and Accreditation Council member too. Madam has presented more than 60 papers in various national and international seminars and conferences. Madam has visited USA and UK for paper presentations. Madam also is on the editorial board member for many peer-reviewed journals. With this brief note, I am honored to welcome Dr. Sridevi Madam for today's session. Over to you, Madam.
Thank you for the nice introduction. Shall I share my screen?
I'll make you the host, Madam. 1 second.
Okay.
Yes, Madam, you're the host now. Madam.
Okay. So the last share was shared first. Okay. Is it visible?
Yes,
Yes,
ma'am.
Madam, it is
Okay.
visible, Madam.
Okay. Shall I proceed then?
Yes, Madam, if it is in presentation mode, it would be good. Madam.
Okay, I'll do that.
Yes, Madam. Thank you, Madam.
Now is it okay now?
Yes, Madam.
Okay. Thank you once again for APP President and organizers for giving me this chance to share my research knowledge with you. And today I would like to present the phytochemical and pharmacological evaluation of Jasminium auriculatum for nephroprotective activity. So these are the contents that I am going to be cover. So the first, the introduction. So nephrotoxicity. So the nephrotoxicity means it is nothing but the other name for this is a renal toxicity, and this renal disease may be caused by direct or indirect exposure to the medicines or the industrial or environmental chemicals, and a number of therapeutic, not only the industrial chemicals and the other things may cause, but even the therapeutic drugs such as the paracetamol, cisplatin, and doxorubicin, all these will causes the kidney toxicity. And this nephrotoxicity may be two types, that is acute and the chronic. And what are the symptoms of these nephrotoxicity? And the symptoms, when we come to the symptoms of this nephrotoxicity, the first thing is it affects the glomerular filtration rate, that is it reduces the urine. And the second thing is it inability to kidney to excrete the metabolites. The major function of the kidney is to expel the waste compounds, water soluble waste compounds from our body. So whenever a person is suffering with a renal problem, so he inability to excrete the metabolites. And the third one is failure in the regulation of the electrolyte balance. As the water is not excreting in a proper way along with the waste or the toxic compounds, generally there is an irregularity in the electrolyte balance may be observed. Then what are the risk factors of this nephrotoxicity? So how it may causes the nephrotoxicity? The first thing is, so multiple medication that can cause the nephrotoxicity. So aminoglycosides such as the gentamicin will causes the nephrotoxicity, and cisplatin. And not only this, long-term usage of non-steroidal anti-inflammatory drugs will also causes the nephrotoxicity. And apart from that, the smoking will also causes the nephrotoxicity, and overweight or the obesity is the another cause for the nephrotoxicity. And other reasons for the causing of nephrotoxicity are the exposure to the chemicals like ethylene glycol, carbon tetrachloride, sodium oxalate, and the heavy metals. These are the other risk factors which led to the nephrotoxicity. And what are the evaluation methods of this nephrotoxicity? There are in vitro and in vivo methods are available. These are the in vitro methods. And the next thing is in vivo methods. In vivo methods includes the estimation of clinical parameters such as blood urea nitrogen, serum creatinine, urinary total proteins, and urinary creatine, or the in vivo methods. And the other one is glomerular filtration rate, then the tubular reabsorption parameters, and the body weight is also considered as the one of the factor to calculate the nephrotoxicity. Then the next thing, now although a number of nephrotoxic agents are available, why the cisplatin has taken? The first and the foremost important reason is cisplatin is a very popular anti-tumor drug. That is, and majority of the cases, this cisplatin is used to treat the various types of the anti-tumor conditions. And the second thing is, with a single administration of an cisplatin will lead to an nephrotoxicity. These are the two things so which moved us to concentrate on the cisplatin induced nephrotoxicity. So here this is the structure of cisplatin, and the HIPAC name is cis-diamino-dichloroplatinum is its HIPAC name. And this cisplatin can be used in various cancers such as testicular cancer, ovarian cancer, cervical cancer, and head cancer. And although it is a very, very good anti-tumor agent, but its clinical use is limited because of its side effects. The notable side effects, not only the side effect, it is a notable side effects. So such as ototoxicity, gastrotoxicity, sensory neuropathy, and hematogenicity, and the most importantly, nephrotoxicity. Okay. So although the number of persons who are suffering from the cancer is increasing from day by day, and the uses of the cisplatin to treat the nephrotoxicity to treat the cancer is also increasing. So what happens is, whenever they are using the cisplatin, the side effect is nothing but the nephrotoxicity. So the researchers have continuously searched certain substances which will provide the nephroprotection against the cisplatin. But till date, there is no drug is available in allopathy to treat the nephrotoxicity. So researchers were concentrated on the alternative system of medicine such as the natural products, and the literature survey is also demonstrated that the number of medicinal plants played a important or the crucial role to attenuate or reduce the nephrotoxicity which is induced by the cisplatin. So based upon literature and the phytochemical and ethnobotanical data of the roots of the Jasminium auriculatum, we were selected to screen the nephroprotective activity against the cisplatin-induced nephrotoxicity. So this is the taxonomical classification of our Jasminium auriculatum, which belongs to the class of Foliaceae. And regarding this picture will shows the roots, okay, root part of our Jasminium auriculatum. And then coming to the vernacular names. So in English, this is called as an Indian Juhi, and in Telugu, it may be called as an Adavimalle, and Kannada, it is Sannamalle, and in Tamil, UC Malagai. Then certain claims are also present in the standard books of regarding this plant. The roots of this plant is used for the ringworm infections, eye diseases, headache, leprosy, mouth ulcers, renal calculi, burning maturation, and wound healing. And flowers of this plant is also claimed for the cardiotonic, diuretic, and ulcer, stomatopathy, and urolithiasis, nephrolithiasis, and dermatopathy. In all these things, there are claims. And apart from that, certain reports, certain research work has also been done on this Jasmium auriculatum. And the leaves of this plant is used as reported for the diuretic activity, in vitro, and antioxidant activity. And apart from that, it is also reported for the immunostimulatory activity. Then regarding the flowers were reported for the anti-lithiatic activity, and the aerial pods were reported for the antibacterial activity. So there were no reports till date on the roots of the Jasmium, although it is having so many claims, but till date there were no reports. That is the one reason to take up this. And the second thing is, certain phytoconstituents are also reported in the Jasmium auriculatum. The leaves were reported for the presence of lupiol and limonene and phytol, which belongs to the terpenoid family. And apart from that, the aerial parts consisting of a phenolic compound. So when you observe these two, so the pharmacological activity was done only leaves, flower, and aerial part, and phytochemical studies were done only on the leaves and the aerial part. So there were no single report on the pharmacological activity as well as the phytochemical studies on this Jasmium auriculatum. So this moved us to do the activity because it is also having the renal calculi activity. So because of that reason, so we are concentrated, or we moved our study towards the screening of nephroprotective activity of Jasmium auriculatum. So the objectives of our study is to conduct the pharmacognostic studies. This is very, very important. So before going for the phytochemical studies or the pharmacological studies, first we have to see that whether any pharmacognostic studies are done or not, which gives the authentication of the plant, whether we are taking the right plant or not. Okay, it gives a purity of the plant. So there were no reports even on the pharmacognostic studies also. So we initiated our work with a pharmacognostic studies of roots of the Jasmium auriculatum. Then the next objective is to prepare the ethanol extract of the roots of Jasmium auriculatum and to conduct the preliminary phytochemical studies. Later, we moved to evaluate the nephroprotective activity of ethanol extract of roots of Jasmium auriculatum. These are the three objectives. So the plan of work is, the first thing is collection of the plant material. So we are collected the roots of the Jasmium auriculatum from the Talakona forest of the Chittoor district, Andhra Pradesh. And after collecting, we were washed thoroughly under the tap water, then shade dried, then it was authenticated with a authenticated person by the Dr. Madhava Shetty, botanist, herbarium keeper of the SV University. So he was authenticated, and a specimen copy was also deposited in the botany department of the SV University. And after that, we subjected it for the shade drying and powdering. We were powdered and then moved for the powder microscopy and the fluorescence analysis and physicochemical properties of that root. Then the simultaneously next what we are done is we are taken the powder and we are prepared the ethanol extract by hot extraction method. Here what we are done is we are taken the plant material and macerated it with a rectified spirit for 24 to 48 hours. Then it was heated on a water bath for 3 hours. Then filtered. The filtrate was collected and again the mark is treated with an rectified spirit and the procedure was continued until thrice. And the filtrate that was obtained in the 3 times were combined and concentrated under reduced pressure to obtain the concentrate of the ethanol extract, and it was stored in the desiccator until it was used. Then next we followed for the preliminary phytochemical studies, and this was done by the conducting the chemical tests, which we were followed in the standard books procedure for the presence of flavonoids, steroids, tannins, saponins, and other phytoconstituents by the respective chemical tests. Then after conducting that, we are also followed the TLC studies based upon this preliminary phytochemical studies. Then we moved for the nephroprotective screening of the ethanol extract. Then for this we will see next regarding the pharmacological activity. So the experimental animals that we were taken is the male albino rats, which are weighing in between the 150 to 200 grams, and they are fed with a standard rodent pellet, and all animal experiments were conducted according to the CPCSCA regulations. And the next thing is before proceeding for these pharmacological studies, we were taken the permission from the IAC of SPMVV, and this is the approval number. Then we were performed the acute toxicity studies according to the OECD guidelines 423. Then based upon these acute toxicity studies or based upon these LD50 values, we were fixed the dose 200 and 400 mg per kg body weight for the ethanol root extract Jasmium auriculatum for the evaluation of nephroprotective activity. Then next coming to the treatment schedule. This treatment schedule we are followed according to the standard journals. Okay, and we divided the animals into 5 groups, and it's a 10-day protocol. And the first group is a normal control, which received only the vehicle from day 1 to day 9. And the second group is a disease control. So where it is treated from 1 to 9 days vehicle. On 5th day, we are treated with a cisplatin at 6 mg per kg body weight. The single dose only. On 6th day, 5th day, we are given the cisplatin. Okay. Then next coming to the lower dose treated animals. So we were treated 200 mg per kg body weight of ethanol extract from day 1 to day 9. On day 5, we are again treated with an cisplatin at 6 mg per kg body weight. And group 4 is served as a high dose treated group, where the animals were received the 400 mg per kg of ethanol extract from day 1 to day 9, and cisplatin is received only on the day 5 at the dose of 6 mg per kg body weight. And here we are taken standard as an cystone. Cystone is a marketed preparation which is available to treat the kidney problems. So because of that reason, we are taken cystone, and this was treated from day 1 to day 9. Then on day 5, we are given again the 6 mg per kg body weight. So until 9 days, we are treated. Same treatment that we are followed, and 9th day after giving this extract or the vehicle, we placed in the metabolic cages for 8 hours, 8 to 8 a.m. to 8 p.m. and given the water and to collect the urine. And 10th day, we are sacrificed the animals by cervical decapitation. Then we are collected the blood by cardiac puncture and sacrificed the animals and collected the kidney for the antioxidant studies in the kidney. Then in the blood, we have performed the biochemical parameters like blood urea nitrogen, and the method is diacetyl monoxide method, and serum creatinine by Jeff's method, and urinary total protein by turbidity method, and urinary creatinine by Jeff's alkaline picrate method. All these methods we are performed by using the HERBA kits. Okay, HERBA kits, and this was performed in semi-automatic and auto analyzer, which is available in our department. So we are performed all these things. These are performed in urine and these are performed in the serum. Okay. Then the kidney tissues were homogenized, and that homogenized kidney tissues we are performed the lipid peroxidation, glutathione, reduced glutathione, and superoxide dismutase and catalyzed enzyme estimation study. Okay. Then all the data has been expressed in mean, standard, error, and the determined by one-way ANOVA variance, and the probability was set at 0.05. Okay. Then regarding the results and discussion. So here the macroscopic study, the first, as soon as we are taken the root, that root we are observed. The color of the root is light brown color and it is odorless, taste is bitter, and the shape and texture is brown color cork is present on surface. Okay, and which contains the slight hairs. And then the pharmacognostic studies, these pharmacognostic studies that we are performed with the help of an expert in pharmacognostics who is available in Chennai. Under his supervision, we are done all these things, and according to him, he has given certain slides, and he explained that this microscopic examination, it contains the root hair. The root is consisting of root hair and 3 to 4 layers of the cork, and followed by the colon-chymatous cells and vesicular bundles containing the xylem and phloem. Okay, so these are the things that are present. And next, coming to the fluorescent analysis of this powder was given. So here the powder was taken and it was treated with the various reagents and observed the color change at the daylight and at 2 wavelengths. Okay, that is UV and the visible light, that is 250 and 365 nanometers range that we are observed. And when the root powder as such, it has a light brown color in the UV light, and the visible light, it is a light brown also. And when they are treated with various reagents, it was shown the different colors. These are the colors which helps to identify the purity of the compound. Then the physicochemical analysis of the root of Jasmium auriculatum was also performed for the extractive values and the ash values. So here the total ash value is 3% and acid insoluble ash is 0.99% and water insoluble ash is 1.2%. So I didn't explain how we are performed is because all these are the standard procedures which are available in the standard books. Okay. All standard pharmacognosy books. That's why I'm not explaining all these things. And extractive alcohol soluble extract is 20% and water soluble is 17% and chloroform soluble is 40%. And the loss on drying that they are observed is 9.49. Okay, the moisture content otherwise. And the next coming to the, we are all conducted the preliminary phytochemical studies of the root powder of, ethanolic extract of Jasmium auriculatum. Except the alkaloids and amino acid, it was given the positive test for all the phytoconstituents. These are the respective tests that we were performed. And thin layer chromatography was performed for the phenolic compounds, flavonoids, and glycosides. Okay. So as this solvent system the mobile phase was taken based upon the standard TLC book, that is Stahl. Okay, TLC by Stahl. So from this book, we are taken the mobile phase or the solvent system, which is suitable for the phenolic compounds and the flavonoids and the glycosides. And for the phenolic compounds, when we are run in a thin layer chromatography, it was observed as two spots. The RF values is 0.27 and 0.36. And flavonoids, it was observed the one spot, it is 0.87. And for glycosides, it was observed that 3 spots, 0.91, 0.75, and 0.27. Okay. Then regarding the pharmacological activity. So here, the first thing that we observed is the body weight of the compound. And the body weight was observed 3 days once, and initial weight that were taken and final weight that we are taken. And when you observe this, when the group 1 is in a normal group, here the body weight was increased. In group 2, it was treated with only the cisplatin. So when you observe this, the body weight was reduced. So the body weight was reduced in the group cisplatin treated, only cisplatin treated group when compared to that of the normal group. And here in the 3 and 4th groups, they received the extract lower and higher dose received groups. Here the change, the body weight is reduced, but the reduction, the percentage of reduction is very less when compared to that of the group 2. So here the comparison was done. The group 2 is compared with the group 1. And group 3 and group 4 were compared with the group 2. Okay. And here the acetone is also compared with the group 2. Okay. Then the next thing is effect of this ethanol extract of Jasmium auriculatum. EEJA means ethanol extract of Jasmium auriculatum on serum parameters of cisplatin induced nephrotoxicity. Not only the serum biomarkers, but also the urinary functional parameters. Okay. Here, when you observe that in group 2, the levels of the serum creatinine is increased when compared to that of the group 1. And in group 3 and group 4, it is dose dependently reduced when compared to that of the group 2. Okay. Likewise, same was observed. The blood urea nitrogen levels were increased here, and here it is almost coming to the normal when compared to that of this. Okay. Then urinary creatinine. Here the levels of the urinary creatinine is reduced. The excretion levels are reduced when compared to that of the normal group, but urinary creatinine is increased here. So it is an indicative. So the blood urea nitrogen should be reduced because the urea nitrogen should be go out. Okay. And serum creatinine should also be go out and it should be reduced. And it should be in urine, the creatinine should be excreted. So when it is excreted, then only it is reduced. If it is not excreting, it will increases. Likewise, here the urinary creatinine levels were reduced when compared to that of the normal control. And the group 3 and group 4, which were received the extract, they are shown the increased levels of the urinary creatinine when compared to that of the group 2. And in case of total urinary protein, the protein should not excrete through the urine, but due to the cisplatin treated group, some renal damage will occur. Due to that reason, the proteins will be excreted. So the total protein excretion is high when compared to that of the normal group. And the group 3 and group 4 were shown the dose dependent protection. You just see the levels were, when you see this, somewhat okay reduced. Then like this, it was showing that. So same in case of the antioxidant enzymes such as catalase, SOD, glutathione. Okay, these 3 antioxidant enzymes in the control levels were in case of the catalase, SOD and glutathione, the control levels are very high when compared to that of the cisplatin treated groups. And the group 3 and group 4 animals which received the extract along with the cisplatin were also increased the levels of catalase, SOD and the glutathione. And regarding the MDA, that is lipid peroxidation or the malondialdehyde levels, here the malondialdehyde levels were reduced in the normal control, and it is very high in case of our group 2 treated groups. And in case of our group 3 and group 4, it was somewhat reduced when compared to that of the group 2. So in total, what is the summary that we all learned today is the pharmacognostic studies that we are performed, which helps to the purity and the identification of the plant material for the future researchers. And by conducting the preliminary phytochemical studies, we came to know that the compound is consisting of the various phytoconstituents such as flavonoids, terpenoids, tannins, carbohydrates, and glycosides. And the TLC studies that we performed was given the presence of flavonoids, and it is further supported for the presence of flavonoids, terpenoids, tannins, carbohydrates, and glycosides. And screening of nephroprotective activity, it was shown that when the animals were treated with an extract, that is ethanol extract of Jasmium auriculatum, it was ameliorated the renal toxicity. This may be caused because of the alteration in the alteration in the mechanism that may be induced by the antioxidant principles which are present in the plant. Okay, this may be the reason why they are showing the nephroprotective activity. There are various mechanisms that they were proposed for the cisplatin induced nephrotoxicity. That may be the release of the inflammatory factors, and the changing, damaging on the layer of the glomerular filtration or glomerular layer, which alters the glomerular filtration, and also releasing of the free radicals. All these are the mechanism of action of cisplatin, but till date they are not. These are proposed ways, but they are not confidently known that this is the mechanism by which the cisplatin will induce the nephrotoxicity. But free radical release is one of the way by which it will shows the free nephrotoxicity. So to stop the release of the free radicals, so the plants or the material, the substances should have an antioxidant principle which may reduces or the control the nephrotoxicity. That may be the one of the proposal. So our plant is also consisting of the free antioxidants principles such as flavonoids, terpenoids, and tannins, which are rich. So the plant is rich in antioxidant principle. Due to that reason, it may shows the ameliorating effect on the renal toxicity induced by the cisplatin in dose-dependent manner. And in conclusion, the current study validates the ethnomedicinal value of Jasmium auriculatum against the cisplatin-induced renal toxicity. So the claim, so the tribal people are using this for the treatment of renal calculi and nephrolithiasis, but there were no proofs, scientific proofs. This is one of the report which will validate the ethnomedicinal use of Jasmium auriculatum. So these are the references, and thank you.
Good afternoon, ma'am. It's Dr. Chennapati.
Good afternoon, sir.
So whatever your presentation is very nice, ma'am. You explained it all the things.
Thank you, sir.
And whatever the availability, ma'am, is it the Adavimalli is available only specific areas, or it is available?
Actually, it is available, abundantly available in Chittoor district, sir. And especially it is available in Talakona forest, sir, in Chittoor.
Okay. And ma'am, you taken that extract or ethanolic extract. Can you tell me the solubility parameters, ma'am? The powder.
Powder solubility parameters, sir.
Yes, sir.
Yes, sir, ethanol extract solubility parameters. I didn't understood, sir.
Is the root powder, it is soluble in ethanol only or any other things, other solvents?
The powder, sir?
Yes, ma'am.
Okay. So actually it's a powder, sir. It is some extent sparingly soluble in water also, sir, because of presence of water soluble constituents. Okay. Dry powder, I'm saying something about dry powder, sir.
Yes, ma'am.
Okay. Dry powder is soluble in water also, and hydroalcoholic solvents, it is soluble, sir. That is, you have to add the water as well as the ethanol, sir. It is also soluble in that also, sir.
Okay, ma'am, thank you for your presentation. And any questions from the participants?
Madam, I have a small question.
Yes, sir.
Madam, is this phytoconstant is specific for cisplatin-induced nephrotoxicity?
Sunil sir, your voice is very low. Sunil sir, your voice is very
Yeah,
low.
madam, I'm checking my audio. Madam, am I audible now, madam?
Yes, sir. Yes, sir. Yes, sir.
Madam, is
But
this
voice is low. You are audible.
Is this phytoconstant only for cisplatin-induced or any metal complex-induced nephrotoxicity, or it can be used for any other drugs?
Sir, actually, the cisplatin, we are tested for cisplatin-induced nephrotoxicity, sir. Okay. The mechanism of action of nephrotoxicity induction by the cisplatin may be different for the heavy metal induced mechanism. If the heavy metal induced mechanism involves the free radical release, then it may also used for that purpose also, sir. Okay, we are not studied for that. It can be extended. The research can be extended for the that also, sir.
Yeah, thank you, madam. Thank you. Thank you very much,
madam. Dear ma'am.
Hi.
Yeah, so to all the listeners of this session, I am proudly expressing myself that I am the student of Sridevi madam, and she is our first professor in Padmavati Mahila Vishwavidyalayam, first appointed professor of the entire pharmacy session. So the most significant part it is. So we are the first batch from Padmavati University. So thank you so much, madam, for accepting President Sir's invitation and being with us today. And so no comments, ma'am, because we are. I still feel I'm kid in front of you. So all other my other colleagues, they expressed. So thank you so much, ma'am, for spending valuable time. And we hope you will continue the same rapport with us, the St. Paul's College of Pharmacy also in future. Thank you, madam.
Definitely. Thank you, Kiran Bhai. Thank you very much for your affection and
Thank you, madam, for the nice lecture.
Thank you, sir.
And just if when I gone through the literature, I have seen some of the herbal drugs. Those are inserted into the face masks. So, are you aware of those? Any of the drug, or you can elaborate that? And I have seen one published paper on that actually, that where herbal masks strategy was used. If anything in detail, if you can elaborate, please.
Actually, sir, we are not worked on that level, but the papers are there. So some of the, not only for the mask, even for the sanitary napkins also, certain antibacterial containing herbal compounds, extracts or compounds are inserting in that sanitary napkins and they are using. That is the one thing I have gone through the research papers. And the second thing, recently what you have said. So this, what do you call this? These masks are imbibed with the herbals. So herbal compounds which are having the antiviral and antibacterial property. So they are taking the, what do you call this? Some fibers, and those fibers, they are macerating in the what do you call this, herbal extracts which are rich in antibacterial and antiviral, and allowing them to drying, and by following the standard procedures for the manufacturing of the masks they are following, sir. But I'm not in depth, I didn't study that, but I have to, sir, because it's a need of our. So I have to do that. I'll do that.
Thanks for the update, Professor Devi.
Thank you, sir. Any other?
Thank you so much, madam, for your wonderful session and for your time for today's session. Indeed, it is very helpful for us, and thank you so much, madam, for your time.
Ma'am, my personal request to you is please join with us from 2 o'clock for valedictory ceremony also, madam, if possible.
Okay, I'll join. I'll join. Thank you for your invitation, Kiran bhai. Thank you.
So it will be at 3, na? Not 2?
2 o'clock, no, sir. So you please tell sir now. As per that, we'll take little more break, sir, because we need to compile all the winners list, no, sir. We need little more time,
no,
sir. Shall we
as per
start at
the
2:30, sir?
No, actually, there are other national award announcements. They will enter at that time only. So we can't change
2
the
only,
time.
sir. 2 o'clock sharp,
Three.
sir.
No, it is at 3 to 4:15.
3 to 4:15.
Yeah.
Okay, sir.
Thank you.
Okay, sir.
Okay. Okay, sir. Shall I remain? Shall I remain? Thank you. Thank you, APP President and the organizers, especially the Kiran Mai and Chandrasekhar, for giving me this chance. Thank you once again. Thank you.
Thank you, madam.
Thank you. Shall I remain?
Harita, please announce the next session timing and all.
Yes, madam. All the delegates are requested to join us back with the same link at 3 o'clock in the evening, IST. At 3 o'clock, we are going to start with the valedictory session. And in the valedictory session, we are going to announce even the prizes of oral and poster presentations of the students as well. See you all back at 3 o'clock in the evening. Thank you, everyone.
Water. Hello.
Hello.
Hello. Yeah, this is Tilak. Yeah.
Hello, sir. Good afternoon, sir. Welcome to the session.
Good afternoon.
Yes, sir. We'll start it by 3 o'clock, sir.
Sure, sure. No problem.
Thank you. Good afternoon, sir. Rajiv sir, shall we start, sir?
Yeah, it's okay. We can start. I'm ready.
Yes, sir. Yes, sir. Madam. Thank you, sir. Kiranmai, madam, shall we start the session, madam? So in just 1 minute, we will start. Sir, madam is joining.
Okay.
Thank you. A very good afternoon, one and all. To improve the golden moment of opportunity and catch the good that is in our reach is the great art of life. A very warm welcome to the galaxy of intellectuals, invited guests, faculty members, and students from different pharmacy colleges. I, Mrs. P. Nagaharita, Assistant Professor from the Department of Pharmaceutics, immensely pleased to present before you and offer a gracious welcome to you all for the valedictory ceremony of 17th Indo-US Virtual International Conference on Recent Advances and Global Trends in Pharmaceutical Sciences, organized and hosted by St. Paul's College of Pharmacy, Turka-MJL, Hyderabad, in collaboration with Association of Pharmacy Professionals, Karnataka State Branch, APP American International Branch, and APP Analytical Chemistry Division. I welcome you all once again to the valedictory session. And to continue with the valedictory session and further announcements, I hand over the session to Dr. Rajiv Dahiya, sir. Over to you, sir.
Yeah. Thank you, madam. So I will share the PowerPoint presentation and we will, I'll ask others to endorse the awards. So I welcome all of you in today's award ceremony of the APP 17th Indo-US Conference. So I would like to start from the APP announcement. So we are announcing our new local branch, APP Hyderabad, which will be APP Hyderabad local branch, and it will be led by the Vice Principal of the Hosting Institute, Professor Kiranmai Mandwa. So I congratulate Professor Kiranmai for this new venture, and she can decide on the other members of the branch to start the activities. Another, there is modification in the APP Molecular Pharmacology Division. The national head, old national head, Dr. Gaurav Gupta, has completed the tenure, and new head will be Professor Somnath D, Professor of Pharmacology at the Hosting Institute. In APP Uttar Pradesh State Branch, the new executive member has joined, Dr. Nilesh Kumar Dwivedi, who is principal at Nand Kishore College of Pharmacy, Naini, Allahabad, Uttar Pradesh. So these three are the, one is the new launch and two are the member modification. So first of all, I would like to start of awards of national categories, and these will be at faculty level. So in among this, one is national category, another is international category. So I would like to start with the international category award. So this is Young Scientist Award 2022, and this goes to Dr. Neeraj Kumar Floria, who is Senior Associate Professor at Department of Pharmaceutical Chemistry at Faculty of Pharmacy, AIMST University, Malaysia. So congratulations, Dr. Floria. If Dr. Floria or Mrs. Dr. Floria is there, so they can endorse the award.
Thank you, sir. On behalf of Dr. Neeraj, because he is underwent surgery, so he couldn't join. So I'm most thankful to Regarding for appreciating all the efforts towards the pharmacy profession. Thank you so much.
Thank you, Dr. Floria. So this award is usually given to the professionals who have published well in the pharmaceutical field and achieved an ISI impact factor of above 100. So other applicants are also welcome for the award, so they can also apply for the upcoming years. Another award is National Award, which is Young Talent Award, and this award goes to Dr. Sunit Kumar Sahu, who is lecturer at Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar, Odisha, for his vital contribution in the pharmaceutical teaching and research. So congratulations, Dr. Sunit Sahu.
Thank you, sir. Thank you, sir.
So you can share your views if you want.
Yes, sir. Sir, good afternoon, everyone on this platform. Thank you, sir, very much for considering my name from this recognition. I am really feeling proud and honored on being awarded this prestigious award from an esteemed professional body like APS. I have been associated with this professional body for the last 3 years. During this period, I found APS is doing a laudable job in upliftment of pharmacy professions, starting from the base level to the apex level. In one hand, it is encouraging and upskilling the pharmacy students by conducting several competition at state level, national level, and international level. And on the other hand, it is helping our young pharmacy scholars to resolve their research project issues and enhance their educational outcomes through organizing excellent seminars and conferences. It is a platform which invites world-class scientists to share their experience, expertise, and educate the young professionals. Thank you, Rajiv sir and Sunita madam, for your relentless efforts to 45 pharmacy professionals in educational, research, as well as healthcare sector. I really feel gratified to be associated with such a great professional body. Thank you all again. Thank you, sir.
Thank you, Dr. Sahu, for the compliments and congratulations for the award. So we'll move to the next category of award. So always when we organize the conferences in one state, we invite the best student awards from the other states. So this time we received the applications for 4 different states. So, and those awards goes to So first award goes to Uttar Pradesh state, and the award is with Mr. Aman Singh, who is D.Pharm student of Nand Kishore College of Pharmacy, Naini, Allahabad, Uttar Pradesh, for his vital academic performance. If Mr. Aman Singh is with us, he can share his views. Hello. Anybody from the hosting institute? Hello. Yes, Mr. Aman. Yes.
Sir, my voice is clear.
Yeah.
Very good afternoon, everyone. And hello.
Yes, we are audible. You can go ahead.
Okay. Very good afternoon, everyone. And it is hard to put this feeling into the word. I am very glad to deliver my speech on such important and movement occasion in my life. And a special thank to my principal sir who is giving this opportunity, and a special thank to Academic Association of Pharmacy Professional Samiti. And thank you so much, sir.
Congratulations, Mr. Aman.
Thank you so much, sir.
Next state award belongs to Punjab state, and the award goes to Jasmine Kaur, who is B.Pharm third year student at Department of Pharmaceutical Sciences and Drug Research, Punjabi University, Patiala, Punjab. So congratulations, Ms. Kaur. If you are with us, you can share your views.
Am I audible?
Yeah.
Good afternoon, everyone. I'm honored to receive this award today. I would like to thank APP and President Dr. Rajiv Dahiya sir for awarding me with the Best Student Award. Especially, I am highly thankful to our professor Dr. Bharti Sapra and all my teachers for recommending me. Thank you.
Yeah. Congratulations, Ms. Kaur. Keep it up. So we'll move to the next award. From the Andhra Pradesh state, the award goes to Mr. B. Rahul Naidu, who is B.Pharm student at VGH College of Pharmacy, Rajamundry, East Godavari, Andhra Pradesh. Congratulations, Mr. Naidu. If you are with us, you can share your views.
Thank you so much, sir. A very pleasant good evening to all the members of Association of Pharmacy Profession. Myself, I am Rahul, studying 4th year B.Pharm in VGS College of Pharmacy, Rajamahendravaram. First of all, I thank you for giving me this opportunity to share my experience with regarding to Best Student Award. Firstly, I thank my principal, sir, Dr. Narendra, sir, for pushing me to move forward in academics. Thank you so much, sir, for the great support towards my career, sir. It's hard to put this feeling into words. Nevertheless, I'm very glad to deliver my speech on such an important and momentous occasion in my life. It's a huge honor for me to receive this award for being the best student of the year. I'm deeply honored to my superiors for this recognition of my work. Thank you, sir. Thank you so much, sir.
Congratulations, Mr. Naidu. Keep it up. So
Thank
we'll
you,
move
sir.
to the next state. From Rajasthan state, the last Best Student Award in outer state category goes to Miss Ria Intodia, who is B.Pharm student at Sri Nathji Institute of Pharmacy, Nathdwara, Rajsamand, Rajasthan. Congratulations, Miss Ria. If you are with us, you can share your views.
Thank you so much, sir. It's a huge honor for me to receive this award for being the best student throughout my graduation. I want to thank APP Karnataka branch and St. Paul's College of Pharmacy and my college, the Sri Nathji Institute of Pharmacy. And a special thanks to my principal and my guide and mentor, Dr. R.S. Bhaduria, sir, and Dr. Gunjan Jadon, ma'am. And special thanks to my parents, family, and friends. Thank you so much, Rajiv Dahiya, sir. Thank you so much.
So congratulations, Ms. Ria. Keep it up.
Thank you so much, sir.
So we will move to the next category. APP awards in local category at faculty level. So these awards belong to the hosting institute. So I will announce the award one by one, and anybody from the hosting institute can endorse the achievement of the awardee. So first award is Best Teacher Award, and it goes to Dr. Sunil Kumar Chaitanya. Congratulations, Dr. Sunil Kumar. Anybody from hosting institute can endorse the achievements of Dr. Sunil Kumar?
Yes, sir. Dr. Sunil Kumar Chaitanya, presently working as Professor and HOD Pharmaceutical Analysis, Training and Placement Officer at St. Paul's College of Pharmacy, Hyderabad. Sir has graduated his B.Pharmacy from Sri Padmavati School of Pharmacy, Tirupati, affiliated to JNTU, Hyderabad in 2002. He has done his M.Pharmacy from NGSM Institute of Pharmaceutical Sciences, Mangalore, affiliated to Rajiv Gandhi University of Health Sciences in the year 2005. He received his Doctor of Philosophy in Pharmaceutical Sciences from Rajiv Gandhi University in 2011. Sir, expertise research areas are pharmaceutical organic chemistry, pharmaceutical analysis, and chemistry of natural products. He is having 16 plus years of teaching and research experience in various pharmacy colleges. And sir has published around 32 publications in international and national journals, and also presented around 10 papers in national and international conferences. Dr. Sunil has a unique nature of guiding and creating enthusiasm in farmer students right from the beginning of his career. He has helpful and amicable nature and won the hearts of many students.
Congratulations, Dr. Sunil Kumar. If you are with us, you can share your views.
Good afternoon. Sir, am I audible?
Yes, sir,
Yeah.
you are audible, sir.
Firstly, I would like to thank APP for selecting me for this nomination, and my sincere gratitude to our college management and for their belief in us. And I would like to share this honor with all my students who have been come across my career. Thank you, sir. Thank you very much.
Congratulations, Dr. Sunil Kumar. Next award in local category is Best Researcher Award, and this award goes to Dr. Vinu Madhav Katla of Hosting Institute, who is Professor of Pharmaceutics. Congratulations, Dr. Vinu. So anybody can endorse the...
Dr. K. Venumadhav, presently working as a professor and head Department of Pharmaceutics and in charge for the mentoring system at St. Paul's College of Pharmacy, Hyderabad. Sir has done his B Pharmacy from St. Peter's Institute of Pharmaceutical Sciences, Kakatiya University, Warangal in 2000. Sir has done his M Pharmacy from University College of Pharmaceutical Sciences, Kakatiya University, Warangal in 2007. He received Doctor of Philosophy in Pharmaceutical Sciences from Kakatiya University, Warangal under the esteemed guidance of Professor Kishan. Sir's expertise research areas are pharmaceutics and novel drug delivery systems, especially lipid drug delivery systems, nano-based systems, etc. He has published 18 research papers in national and international journals and presented 15 papers in national and international conferences. He has attended over 63 conferences, both national and international, and guiding one PhD student and 25 M.Pharmacy students and around 10 B.Pharmacy students. Dr. Venumadhav meant for his calm going and stable professional behavior and committed personality.
Okay.
Congratulations, Dr. Venu, sir.
Dr. Vinu, you are with us?
Thank you, Dahiya sir. Good afternoon to all. Firstly, I want to thank APP US and Karnataka State Branch for selecting me to this award. Next, I would like to thank the management, principal, and all my dear colleagues of St. Paul's who directly or indirectly being a part of my research journey. And lastly, but not least, all my M.Pharm students. Thank you once again.
Congratulations, Dr. Vinu. Next category is Best Achiever Award, and the award goes to the Vice Principal of the Hosting Institute, Dr. Kiranmai Bandwa, in recognition of her noteworthy teaching and research services. Congratulations, Dr. Kiranmai.
Thank you, sir.
So you can share your views.
Yeah. Dr. M. Kiranmai ma'am, presently working as Professor and Vice Principal Accreditation in charge at St. Paul's College of Pharmacy, Hyderabad. She has done her B Pharmacy from Sri Padmavati Mahila Vishwavidyalayam, Tirupati, and M Pharmacy from JSS Academy of Higher Education and Research with distinction and is a gold medalist. She has received Doctor of Philosophy in Pharmaceutical Sciences from JNTUH. She is having 18 plus years of teaching and research experience in various pharmacy colleges. She is a chairperson for scientific session Percept Usmania University. She acted as a session chair in International Symposium on Medical Therapy Management organized by TMED Research Solution in association with IPA on 30th November 2021. She was chaired at Avenida International Conference 2021. She has an expertise in research areas like medicinal and green chemistry, nanomedicines, and multidisciplinary research, and also metallic nanoparticles and their applications. She has got a patent filed on an antimicrobial composition and its preparation method. She has 56 international and national publications. She has attended international national conferences and presented papers and also received awards. She has Google Scholar citations of around 510 and Scopus citations of around 200 plus and has H index 11. She has a good experience as an administrator. She is proficient at guiding projects for UG and PG students of around 15 plus UG projects and 25 M-Pharmacy projects and 5 PharmD projects. She is experienced in establishing well-equipped laboratories. She was a guest speaker in the International Online Scientific Congress, Phytomed 2020, organized in Melbourne, Australia. She has also published a book chapter titled as Fabrication and Functionalization of Other Inorganic Nanoparticles and Nanocomposites, Taylor Francis Publishers, 2019-20. Recently, ma'am has received Scientific Excellence Award 2021 by Academic Growth Foundation 2021. Under her flagship as an accreditation in charge, St. Paul's has accredited by NBA. A great inspiration to all of us. Under her leadership, we gained the confidence of winning, not only winning, but excelling from her part. As a leader, she always accepts challenges, faces them, and conquers them successfully. We congratulate you, ma'am.
Thank you very much.
Thank you, Dahiya, sir. Thank you so much. Am I audible, sir?
Yeah, you are.
Yeah, yes, sir. So whatever my colleague read about me, so that is only beginning. I feel like I want to be the constant learner, sir. I want to learn many things from elderly people like you and many other eminent personalities of pharmacy profession, because we should not stop with small achievements, I feel. And really it's challenging period for me from last 2 years. So facing the pandemic obstacles and dealing with many multitasking issues in my institution, but by God's grace and with the immense support of my management, Sudhir Garu and Chairman sir and Secretary sir, Raghav Reddy sir and my staff and all the total team St. Paul's, so they supported me like anything, and giving guidance is different, receiving guidance is different. So both, if they both are balanced, only success can be achieved, I feel. Thank you so much for giving me opportunity. And so for this award also, sir, thank you once again.
Congratulations, Dr. Kiran. So we will move to next category. So I will ask the hosting institute person who are receiving to keep them short with major achievements.
Yes.
Thank you. Best Pharmacist Award. So we have giving 2 Pharmacist Award. One belongs to departmental category, and the award goes to Dr. Afroz, who is Assistant Professor of Department of Pharmacy Practice at the Hosting Institute. Congratulations, Dr. Afroz. So anybody can endorse Dr. Afroz public service.
Dr. Samreen Afroz is presently working as Assistant Professor at St. Paul's College of Pharmacy. She has completed Doctor of Pharmacy, PharmD, from Jawaharlal Nehru Technological University, Hyderabad, with a distinction 75%. And she has gained experience as Senior Research Scientist in collaboration with the projects of Entasis Therapeutics, Boston Biohub, Waltham, USA, and also Bharat Serums and Vaccines Limited, Navi Mumbai. She has 2 publications of international honor, and she got awards as honored as COVID-19 Warrior Award from Remarkable Service in the Department of MICU at Care Hospital in 2021, and represented India in Entasis Therapeutics Summit at Swisstal Merchant Court, Singapore. As a active team member, she served as a COVID-19 team member for Critical Care Medicine Unit in Gandhi Hospital, and she organized Drug Information Center at Gandhi Hospital. With her professional excellence and frontline warrior service serving the society as a remarkable contribution for your unsacrificable services as pharmacist. Congratulations, Dr. Samreen.
So, Dr. Afroz, you are with us. If you want to share the views. Okay, we can move next. So the second award in the Best Pharmacist Award category belongs to drug store category, and it goes to the
Good evening, everyone.
community pharmacist. Who is there? Okay, Dr. Afroz, you can go ahead.
Yes, sir. Good evening, everyone. I really appreciate your words and Sneha ma'am's words for me. It's a great pleasure and great privilege to be here. I would like to thank the creator, my parents, and everyone who has been a part of my journey till date. And I thank Chairman Sir, Sudhir Sir, Principal Ma'am, Kiranmai Ma'am, and all the St. Paul's HODs, Somnath Sir, and all rest of the HODs and faculty members of St. Paul's College of Pharmacy for being very cooperative and understanding me and honoring me for this one. I really thank everyone for this.
Congratulations, Dr. Afroz.
Thank you so much, sir.
So the second, the Best Pharmacist Award, which belongs to drug store category, and it goes to Mr. Tilak Sairam Gajendra for his noteworthy public service. Congratulations, Mr. Tilak. If you are with us, you can share your views, and anybody can endorse the activities.
Tilak Sairam Gajendran, founder of Medox Pharmacy. He has worked as a senior application specialist at electronic medication management at eHealth Queensland. And he has also played a role as a consultant technology at Deloitte USA Consulting Private Limited. He is also a implementation lead pharmacy module at OHM Healthcare Solutions Limited. He also worked as a clinic and dispensary pharmacist at Queensland Health, Queensland, Australia. He has achieved and designed and implemented a guideline for nursing staff regarding the flow of discharge and the leave planning process with respect to the medication dispensing, which streamlined the dispensing of discharge and leave medication delays from pharmacy department. He has managed 35 opioid drug rehabilitation patients for a period of time, 1 year. He has worked as an oncology pharmacist managing chemotherapy protocols for a period of 1 year. He also worked as pharmacist in charge at Hospital Pharmacy Services, Government of South Australia. He has also worked as an architect of the pre-operative antibiotic usage guidelines for surgeons at Waila Hospital, Australia. He has also worked as intern pharmacist at Seoul Pattison Chemist, North Territory, Australia. We congratulate you, sir.
Yes, Mr. Tilak, you are with us?
Yes, I am. How do I turn it on actually?
Yes, you are audible, Tilak. Please go ahead.
Yes. Okay. So yeah, thank you very much for the award. I really appreciate that, and I thank my ma'am for recommending me, and also thank the president of APP. Thank you very much for the award, and look forward to serve the society and much more with a more best practice, clinical practice in the retail pharmacy situation. So I really appreciate for the award given to me, and thank you very much for that.
Congratulations, Mr. Tilak, for your noteworthy public service, and keep it up.
Thank you, sir.
So next category of award, Best Talent Award, and it goes to Mrs. Haritha, Assistant Professor of Department of Pharmaceutics of the Hosting Institute. Congratulations, Mrs. Haritha. So anybody can endorse the CV?
Yes, sir. P. Nagaharitha, working as Assistant Professor in St. Paul's College of Pharmacy, Hyderabad since 2012. She has done her B Pharmacy from Sri Padmavati School of Pharmacy, Tirupati, and M Pharmacy from Rao's College of Pharmacy, Nellore. She has got registered her PhD from Annamalai University. She has of 18 publications and attended 32 conferences and presented 2. She has attended 14 faculty development programs. She has completed 9+ years of teaching experience and a long-standing faculty at St. Paul's College of Pharmacy. She is a very sincere, disciplined, excellent, and enthusiastic teacher. She successfully accomplishes any task given within a scheduled time. We congratulate you, ma'am.
So, Mrs. Haritha, you are with us?
Yes, sir. Thank you so much. Thank you so much, Navya madam, for your sweet words. And I thank my organization, St. Paul's College of Pharmacy, our principal, Dr. Chandrasekhar, our vice principal, Dr. Kiranmai Madam, as well as APP Association, and also special thanks to Somnath sir for identifying the thing that I'm doing. And it is really an encouragement for me to participate and to organize many more events. And I personally thank each and everyone who are behind my success. And I also take this opportunity to welcome our chairman, sir, who has joined the session, Mr. Sudhir, sir. We welcome you, sir. Thank you so much, sir. Thank you so much for the award.
Okay, congratulations, Mrs. Haritha.
Thank
So we'll move to the next category. Best Performer Award, and the award goes to Miss Pratibha, who is PharmD student at St. Paul College of Pharmacy. So anybody can endorse the activities of Miss Pratibha.
Miss Pratibha, she's presently an intern student of PharmD, and she has done her intermediate from Sri Chaitanya Junior Kalashala in 2015. And she has strived all the pace for the betterment of patient care, working in various departments by doing internship in the clinical pharmacy, and conducted various programs to create awareness among the public. She was a chairperson for IPSF St. Paul's chapter from October 2020 to October 2021. And she's also done her internship in Climate Research Solutions from October 2020 to July 2021. And her academic project is assessment of quality of life and evaluation of non-pharmacological management in dysmenorrhea. We present Ms. Pratibha as a young leader from student community, and she is an inspiration for the entire student community. Hearty congratulations, Pratibha.
Pratibha, you are with us to share your views.
Good evening, sir. I'm audible.
Yes.
Yes,
Yes.
Yeah, you switch on your video, Pratibha.
1 minute, ma'am. Am I visible?
Yes, yes,
Yes.
Yes.
Yes, go ahead.
Thank you so much, sir, for giving me this award. Thank you so much, Kiranmai ma'am, and all the faculty of college for recognizing my abilities and encouraging and supporting always to do new things, to initiate new things for the betterment of patient care and also for the betterment of the public health. Thank you so much for supporting all the times. Thank you, sir.
Congratulations, Ms. Pratibha. So we will move to the next category. Best Research Scholar Award, and the award goes to Mr. Peta Sudhakar, who is Assistant Professor of Department of Pharmacology of the Hosting Institute. Congratulations, Mr. Peta. Anybody can endorse the activities of Mr. Peta.
Mr. Peta Sudhakar, presently working as an assistant professor in the Department of Pharmacology at St. Paul's College of Pharmacy, Hyderabad. Sir is registered for PhD in Jawaharlal Nehru Technological University, Hyderabad. He did his Master of Pharmacy from University College of Pharmaceutical Sciences, Kakatiya University, Warangal. Bachelor of Pharmacy from University College of Pharmaceutical Sciences, Kakatiya University, Warangal. And he is having 13 plus years of teaching experience in various pharmacy colleges. His area of research interests are pharmacology and toxicology, in vitro and in vivo evaluation of bioactive compounds in diabetes mellitus and hyperlipidemia. Sir has excellence in guiding the projects for UG students and experienced in establishing well-equipped laboratories. He secured All India 346 rank in GATE 2005 and received UGC scholarship in MPharm. During pandemic, under his supervision, Sir has done adverse drug reaction survey. He has been conducted the ADR survey. Mr. Sudhakar is a wonderful personality, and his Listening skills and commitment towards research or any task assigned made him as a special person. Congratulations, Sudhakar sir.
Mr. Peter, you are with us to share your views.
Yes, sir. Good afternoon, sir.
Yes, good afternoon. Please go ahead.
Thank you so much, sir, for the honoring with this award. Thank you so much, sir.
Okay.
And my sincere thanks to the management for their support and encouragement. I sincerely thank Principal Chandrasekhar sir and Vice Principal Dr. Kiranmay madam, and my sincere thanks to Dr. Somnath sir and all the HODs of the college. And I thank all the faculty for their encouragement and support. Thank you so much, sir.
Okay, congratulations, Mr. Peter. So we will move to the next category of awards. So these are APP awards in local category, which are for students. So the first award, Best Student Award, it goes to BPharm first year student, Miss Talari Anusha. Congratulations, Miss Anusha. If you are with us, you can share your views, or anybody can endorse the CV of Miss Anusha.
Yes, sir. Talari Anusha is presently a BPharm first year student. She is an active student volunteer and won many prizes at national level seminars and scientific model presentations. She is a good performer in academics and also has excellence in co-curricular activities. Being in first year, you are an active performer, Anusha. You have very long way to go. All the best and congratulations.
Miss Anusha, you are here?
Yes, sir. Thank you so much for this award. I hadn't expected this. I thank Dr. Kiranmay ma'am for this and my faculty for my support and everything. Thank you so much.
Congratulations, Anusha.
Thank you, ma'am. Thank you so much.
So the next award in Best Student category for Year 2 student in male category goes to Mr. Sheikh Khalik Ahmed. Congratulations, Mr. Ahmed. Anybody can endorse the activities of Mr. Ahmed?
Yes, sir. Mr. Sheikh Khalik Ahmed is a presently BPharm second year student and a Best Student Award under male category. He is an all-rounder and a very good hard worker. He is a very diligent personality and very disciplined. He is a good performer in academics and always an active participant in co-curricular activities. All the best and congratulations, Khalik.
Mr. Ahmed, you are with us?
Good afternoon, sir.
Good afternoon.
I'm huge honor to receive this award. First of all, I want to thank all St. Paul's College of Faculty. Once again, I thanks to all. Thank you, sir.
Okay, congratulations, Mr. Ahmed.
Congrats, Kali.
Thank you, ma'am.
So in female category for the BPharm Year 2, the award goes to Miss K. Monica Lakshmi. Congratulations, Miss Lakshmi. Anybody can endorse the activities of Miss Lakshmi?
Yes, sir. Monica Lakshmi is presently a BPharm second year student and she is a very good performer at academics and active in co-curricular activities. She has attended seminars including BYJU'S and artificial intelligence and has presented a paper on antimicrobial resistance. She has presented paper at International Symposium on Medication Therapy. In addition, she has a very good motivational attitude. Congratulations for your award.
Ms. Lakshmi, you are with us to share your views. Okay, we can move ahead. Next Best Student Award goes to Year 3 BPharm student in male category, Mr. T. Gadam. Congratulations, Mr. Gadam. We can endorse the CV, please.
Yes, sir. Mr. Tarun Kadam is a presently BPharm third year student. He is a very good performer and has participated in national level webinar on paradox of COVID-19 life conducted by Tiruvallur College of Pharmacy. And he has participated in 1 week webinar on current updates in pharmaceutical research and development conducted by Vishnu Institute of Pharmaceutical Research and participated in international online webinar on Aushadhi Gyan Vikas conducted by AM Memorial College of Pharmacy. Being a third year student, he has bagged a prize in national level e-quiz conducted by Vidya Jyoti Institute of Technology. He is a very good performer and active member in student institutes. And congratulations, Tarun.
Mr. Gadam, you are here to share your views.
Yes, sir. Hello. Good afternoon, everyone.
Hello.
Am I audible?
Yeah, so somebody please mute yourself now. Please don't disturb the program. Preeti, please mute yourself. Yeah, thank you.
Yes, Mr. Tarun. You
Thank
can
our
share
faculty members and our Vice Principal Kiran ma'am for giving good education in the college and encouraging us. Thank you.
Congrats, Tarun.
Thank you, ma'am. Thank you so much.
Okay, we will move to the next award. Next Best Student Award in female category goes to BPharm third year student, Miss Chandana Ganji. Congratulations, Miss Ganji, for the achievement. CV endorsement, please.
Miss Chandana Ganji is a BPharm third year student and she is an active student coordinator member of Swachh Bharat Committee at St. Paul's College of Pharmacy. She has participated in online quiz, that is bio quiz on history of microbiology conducted by Validian Micro Labs and has participated in national level e-quiz conducted by Vidya Jyoti Institute of Technology with a score of 96%. She also participated in national level quiz on general chemistry conducted by Methodist College of Engineering and Technology and participated in online quiz on NSS awareness quiz by NSS unit of Viper. She is an active student member and also a very good performer in academics. Congratulations, Chandana.
Yes, Miss Chandana, you are with us. Okay, we can move to the next student category award. In male category, BPharm fourth year student, Mr. G. Anand. Congratulations, Mr. Anand, for the achievement. CV endorsement, please.
Yes, sir. Anand Gaud is pursuing finally a B Pharmacy at St. Paul's College of Pharmacy with an aggregate percentage of 86 and completed his intermediate from Fortune Butterfly Senior Secondary School with an aggregate percentage of 73.4. He has participated in National Conference in Pharmavriddhi 2020 and also in webinar in skill development for Self-Reliant India by Maharshi Markandeshwar University, Ambala, Haryana, and also participated in national webinar on methods on diminution of polypharmacy among geriatric patients by Seven Hills College of Pharmacy. He has won second prize on working model on digital hospital. He also participated in National Pharmacy Week 2K Run International Inter-College Competition at Hyderabad. And he is also an active student coordinator member at St. Paul's College of Pharmacy. He is a joint secretary at IPA SF St. Paul's Student Chapter, Hyderabad, and also a member and volunteer. I congratulate you, Anand.
Yes, Mr. Anand, you are with us to share your views.
Yes, sir. Yes, sir.
Yes, please. Yes, please go ahead.
Good afternoon to all. A very good afternoon to all. I really thank to APP for giving this award, and I really thank to Dr. K. Kiran ma'am and Dr. Somna sir and Harika ma'am for giving this award. Thank you so much.
Congratulations, Mr. Anand. So we will move to the next award.
Congrats, Anand.
In female category, the BPharm fourth year student award goes to Miss Nikita Kattukuri. Congratulations, Miss Nikita. Endorse the CV, please.
Yes, sir. Kattukuri Nikita, she is a B Pharmacy final year student from the St. Paul's College of Pharmacy
with average percentage of 91%, and she achieved the Best Performer Award in Mahindra Pride Training on Employability, and she received first prize in e-poster presentation conducted by St. Paul's College of Pharmacy in the year of 2021, and she received Merit Award in Intermediate in the year of 2018, and she won runner-up in quiz competition conducted by Global Hospitals in the year of 2016. Congratulations, Nikita.
Nikita, you are with us.
Yes, sir. Thank you so much. Thank you so much. I would like to thank APP and my faculty from St. Paul's College of Pharmacy. Thank you so much. This award will be a reminder for me to giving my best. Thank you, sir.
Yes,
Congrats,
Ms. Nikita.
Thank you, ma'am.
Congratulations. Keep it up. Next award in Best Student Award category. It is Post-Graduation Award in Female category of M.Pharm Pharmaceutics branch, and the award goes to Miss Nabila Begum. Congratulations, Miss Nabila. Endorse the CV, please.
Yes, sir. Nabila Begum is pursuing M.Pharmacy third semester from St. Paul's College of Pharmacy with an aggregate percentage of 8.1, and she has done her B.Pharmacy from St. Paul's College of Pharmacy with an aggregate percentage 6.9. She has completed her intermediate with an aggregate percentage of 9.1, and she has participated in various competitions and won various conferences. She also is a student coordinator of Partnering Institute in ISM TM 2021. She has volunteered in Pharmavidhi 2018, and she received award of Student of the Year in 2015. I congratulate Nabila.
Congratulations, Ms. Nabila. If you are with us, you can share your views.
Good afternoon to everyone. It's a huge honor for me to receive this award for being the best M.Pharm student. And I would like to thank all Chairman Sir, Principal Sir, and all the faculty of the St. Paul's College of Pharmacy for supporting me and guiding me throughout my academic aspects. And also special thanks to Vice Principal and my mentor, Dr. Kiranmay Madam, for supporting me and guiding me throughout the academic research project. And also I would like to thank APP for this recognition. Thank you.
Congrats, Nabila.
Thank you, Madam.
Keep it up. So next award in female categories goes to PharmD fourth year student, Miss Farisa Kojer. Congratulations, Miss Kojer. Endorse the CV, please.
Farisa Kausar, she is a PharmD fourth year student from St. Paul's College of Pharmacy. She has participated in many competitions like conferences, National Conference, Pharmavidhi 2020, organized by St. Paul's College of Pharmacy in association with IPISF, and webinar on the theme of anger and stress management, organized by St. Paul's College of Pharmacy. And she participated in Fresco 2020 national level art competition organized by St. Paul's College of Pharmacy, and she participated in quiz Ozone Day September 16th, organized by NSS team St. Paul's College of Pharmacy, and she is an active member of Pharma Social Hands Together PSHT, registered under Telangana Societies Act 2001. Congratulations, Farisa.
So, Ms. Farisa, you are with us?
Yes, sir. Good afternoon to everyone. I thank our principal, Dr. Chandrasekhar sir, for giving me this opportunity, and APPB for giving me this opportunity. And I also thank my parents for supporting me. Thank you, sir.
Congratulations, Ms. Farisa.
Thank you, sir.
Next award in male category goes to PharmD fifth final year student, Mr. Rizwan Mohammed. Congratulations, Mr. Mohammed. Endorse the CV, please.
Muhammad Rizwan, he is a PharmD fifth year student, and he participated in National Pharmacy Week Celebration 2021 by Sarojini Naidu College of Pharmacy, and he also volunteered in all college professional events. He volunteered in international and national conferences. He's an all-rounder in all the activities. Congratulations, Rizwan.
Mr. Rizwan, you're with us to share your view.
Good evening, one and all. First of all, I feel enlightened to receive this award.
I would like to thank all the mentors and teachers of my St. Paul's College of Pharmacy for helping me to receive this big honor. And I especially heartfully thank all the members of APP to recognize all the efforts of the students and encouraging to achieve much more in life. Thank you so much.
Congratulations, Rizwan. Next award in female category goes to Form D 5th year student, Miss Shivani Deornani. Congratulations, Miss Shivani. Endorse the CV, please.
Shivani, she's the Form D 5th year student from St. Paul's College of Pharmacy. She participated in international conferences, Pharmavriddhi, conducted by St. Paul's College of Pharmacy, and she participated and presented PowerPoint presentations in CVSR College of Engineering College, Ghatkesar, and she also participated in quiz competition in international level pharmacy profession competition conducted by St. Paul's College of Pharmacy in the eve of Pharmacist Day, and she also very quick learner and she's a studious and sincere student. Congratulate Shivani.
Thank you. So next award in female category goes to MPharm pharmaceutics student, Miss Y. Jhansi. Congratulations, Miss Jhansi. Endorse the CV, please.
Ms. Y. Jhansi is pursuing her M.Pharmacy first year at St. Paul's College of Pharmacy. She has completed her B Pharmacy with an aggregate percentage of 75% and intermediate with an aggregate percentage of 98. As she has participated in various conferences, she was selected as Scientific Relation Coordinator by Skynow Healthcare Private Limited. She has served as editor in IPA SF St. Paul's College of Pharmacy. Congratulations, Jhansi.
Miss Jhansi, you are with us to share your views. Okay, we will move to next award. Next award in male category goes to MPharm pharmaceutics student, Mr. E. Parveen. Congratulations, Mr. Parveen. Endorse the CV, please.
Mr. E. Praveen is pursuing his M.Pharmacy in the Department of Pharmaceutics at St. Paul's College of Pharmacy. He actively participates in all the conferences and the seminars conducted by the college and even the competitions that were conducted outside the college. He is a very enthusiastic person and a very studious person who always wants to learn the new things. And he is also an active member of the IPASF St. Paul's chapter, and he is a volunteer in various social activities like Pharma Socio Hands Together, and he is a very enthusiastic student. Hearty congratulations, Praveen.
Mr. Parveen, if you are with us, you can share your views.
Good evening, everyone. First of all, thank you for giving this opportunity, and my heartful thanks to my Dr. Kiran ma'am and Dr. Somnath sir and Dr. Venu Madhav sir, and it is a great encouragement for students like us. And thank you APP for giving this award. Thank you.
Congratulations,
Praveen.
Thank you, ma'am. Thank you, sir.
So next award in female category go to MPharm pharmaceutical analysis student, Miss K. Lakshmi Sravanti. Congratulations, Miss Lakshmi. Endorse the CV, please.
Lakshmi Sravanti is presently a MPharm first year student, and she has completed her B Pharmacy with a percentage of 75 from Vikas Institute of Pharmaceutical Sciences and 82% aggregate for intermediate. She has undergone training in workshop on current aspects and orientations of pharma industry by Pharma Training Institute, and also acting as medical coder in ICD CPT. She has done her project work in BPharm final year entitled with In-Plant Training Report. She's an active student coordinator and also a very good student volunteer and a very good performer in academics. Congratulations, Lakshmi.
Ms. Lakshmi, you are with us to share your views. Okay, we will move to the next award. In male category for M-Home Pharmaceutical Analysis student, the award goes to Mr. Bharat Kumar M. Congratulations, Mr. Bharat. Endorse the chief, please.
Bharat Kumar, he's an M-Pharm
Please, other participants to please mute the audio.
I think we have Miss K. Lakshmi with us. Yes, Miss K. Lakshmi, you can share your views.
Lakshmi, your audio is unmuted. Please stop. Miss Lakshmi Savanti.
Mr. Bharat. So we have read about Mr. Bharat.
No, sir. Yes, sir. Here I'm reading it, sir. Mr. Bharat Kumar is an MPharmacy pharmaceutical analysis student, and he's a topper in academics for the year 21-22. And during his study, he has achieved many of the prizes participating in various level competitions. And congratulations, Bharat Kumar.
So, Mr. Bharat, you are with us to share your views. Miss Lakshmi, if you want to share your views, you can share. She is unable to listen us. You can unmute. Okay, so this completes the local category awards. So before moving to the next categories of award, I would like to congratulate all the hosting college winners. This is we are organizing events since 2011. APP is a 10-year-old body, and it is the first college where we are giving 23 awards in local awards. So I wish to thank the chairman and the principal and the vice principal for giving association the opportunity to honor your students and staff. And we always are with the colleges to honor the students and the staff for their potential. So I really admire that. So yesterday we have conducted the oral competition, and there were 7 presentations. All were very informative, but we have to select for award. There were the announcement of 2 awards. It was a memorial award in the memory of late Mr. Manas Tripathi, and there the first winner will be the winner of Outstanding Oration Award. And there was 1 appreciation award. We have added 1 more appreciation award to that, but that is without the financial prize. So we have kept 1/3 prize, which will contain a medal and certificate. Okay. Second prize, a second appreciation prize, that will be with the prize of Rs 1000 and a medal and a certificate, and first prize, which will be for the memorial award, outstanding oration award, that is with the cash prize of 2000 along with the medal, memento and the certificate. So I will start with from the last one, the third position for the Mr. Manas Tripathi Memorial Award, which will contain a medal and the certificate. And the third position award goes to Mr. Monu Kumar Sukla, who belongs to Sulini University, and he is the research scholar at School of Pharmaceutical Sciences there. If Mr. Monu Sukla is there, he can share his views. Congratulations, Mr. Sukla.
Thank you, sir. Good evening to good afternoon to all. Good evening to all of you. And I would like to thank Association of Pharmacy Professionals and St. Paul's College for organizing such a conference. And I would like to thank President APP, Dr. Rajiv Dahiya sir, Secretary APP, Dr. Sunita Dahiya ma'am, and other dignitaries involved in the conference. And I would like also thank to my supervisor, Dr. Deepa Kumar, for providing me such opportunity to present my work. Thank you, sir, and thank you all. Thank you so much, sir.
So you competed well. It was a close call. So that's why we have to create this additional award for you. So you were very close to the second position holder. Congratulations.
Thank you so much, sir. Thank you, sir.
The second position for this memorial award, the second position holder candidate is Miss Sonu Bedi, who belongs to Amrita School of Pharmacy, Amrita Vishwavidyapeetham, Kochi, Kerala. Congratulations, Miss Beni. If you are with us, you can share your views. This award will carry a cash prize of Rs 1000, a medal and a certificate. Miss Beni, you
Yes,
are
sir.
with us?
Yes, sir. Good afternoon to one and all. I'm so thankful to all of you for this honor. And I would like to thank President and all office bearers of APP and the hosting college St. Paul's for this wonderful opportunity and for this award. And also I would like to thank Dr. Sabidayam, our principal, and my guide, Dr. Anish TP, Associate Professor, Department of Pharmaceutical Chemistry and Analysis, and to all my dear friends, colleagues of Amrita Vishwavidyapeetham, and especially to my beloved parents, my family, all my friends, well-wishers, and above all to the God Almighty. This is the first ever award in my life, and definitely I think it will be inspiring me to achieve a lot in the future. And thanking you all once again. Thanks a lot.
You were very close to the first award winner, but the thing was your studies were, the comment was your studies were on the way. So there you got less marks, and that's why you were on position 2. But you have presented very well. Congratulations.
Thank you so much.
Okay. The first award, which is the Mr. Manas Tripathi Memorial Award, that goes to Dr. Sneha Thakur, who is Associate Professor at St. Paul College of Pharmacy, Telangana. Congratulations, Dr. Thakur, for the achievement. You can share your views.
Thank you, sir. Thank you so much, APP President Dr. Rajiv Dahiya sir and Secretary Dr. Sunita Dahiya madam. And thank you, Kiranmai madam, for creating the opportunity, and Dr. Somnath sir especially. And I'm thankful to my guide, Dr. G. Krishnamohan sir, professor from JNTUH, and also St. Paul's team, and also my family and friends who always supported me and given me this confidence. Thank you, each and everyone. Thank you so much, sir.
Congratulations, Dr. Thakur. But I really have to say it was a very close oral competition. It is all the three what we have given. We have to create a new award, but the points were very, very close. So although we have to decide 1, 2, 3, but I can say all 3 were equal. So all 3 have performed very well, and research was really of international standard. Congratulations.
Thank you, madam.
Congratulations all the 3 winners. So, and the first winner will get the cash prize of 2,000, and our team will contact you in that regard. Congratulations.
Thank you so much, sir.
So we will move to our next thing, appreciation award. So anything will not complete without appreciating the leaders of the hosting college. So association would like to extend the appreciation awards to Mr. K. Sudhir, Chairman and Correspondent, St. Paul College of Pharmacy, who gave us the chance to host the institute, or host the this event at the institute, and given us the chance to honor the students and the staff of the St. Paul College of Pharmacy. Thank you very much, sir. We are also thankful to Professor B. Chandrasekhar and Professor Kiranmai Madam, Professor Somnath D for their hard efforts to create this and accomplish this as a successful event. So this is the certificate appreciation award certificate for Mr. K. Sudhir, and these will be supplied as soon as possible. Also, we extend appreciation certificates to our guests, Professor Madhavbhai M. Patel, who is Vice Chancellor of Sabarmati University, Ahmedabad, Gujarat, Professor Alekha Das, President of our American International Branch from Creighton University, Omaha, Professor Suresh Channupati, our National Head of our APP Analytical Chemistry Division from Telangana. Thanks to our international speaker, Professor Somnath Singh from Creighton University, Dr. Sivkanna Filoria from AIMST University, Malaysia. National speakers, Professor Kuntal Das from Kripanadi College of Pharmacy, Bangalore, Professor S. Satish Kumar from Vistas Wells Deemed University, Pallavaram, Chennai, and Professor Sri Devi from Tirupati Mahila Vishwavidyalaya.
So happy. Make a photo on this, management.
And we extend our special thanks to all the chairpersons and local organizing committee. So now last is the announcement of the best poster award winners and best oral award winners, which the competition for which were conducted at the college. So before switching to the anchor, so I would extend my thank you to all who have put their hard efforts to make this event successful, and hope to meet you in our next event, which will be at Etawah, Uttar Pradesh on 5th of February. It will be our 14th Indo-Malaysian conference, and we will celebrate the World Cancer Day there. So I will, with this, I will switch over to the anchor. Thank you very much.
Thank you so much, sir. Am I audible?
Yes.
Yes, sir. Thank you so much, sir. And now it's the time to announce the prizes for the best oral and poster presentations. So talent and skills are the crops that are nurtured, groomed, and given all possible opportunities to grow and bloom. This is a much awaited event today, as it gives the returns to the efforts put in by the delegates in presenting their works. Before calling in the winners, let me tell you that winning and losing is a part of a game. The most important part is that you took in the challenge to face the competition. So I would say everyone is a winner here. Congratulations to all the participants. And now here we are going to announce the winners of Lubna of B Pharmacy third year. Please mute your audio. Thank you. And coming to the oral presentations, we have the prizes for the department wise. Coming to the Department of Pharmaceutics, the first prize in the oral presentation goes to Reddivari Harika, Research Scholar from KL University. Harika, are you there? And the second prize goes to Dr. C. Mahendranath Reddy, Research Scholar, JSS Academy of Higher Education and Research, Ooty.
వాళ్ళ నేమ్స్ కనిపించట్లేదు హరిత స్లైడ్ షో స్టార్ట్ చేయండి.
Yes, madam, already started the slide show.
No, I can see only first slide here.
Slide show is not working.
No, you didn't share your screen.
Yeah, I'm starting again, madam.
Go for presentation mode.
Is it visible, madam?
No, slide show still it is not in presentation mode. Here you click na down presentation mode.
Yes, madam. Here on my screen, it is showing in the presentation mode.
Okay, what you can do, you can move the slide one by one.
Yes, sir. Share screen. Okay. Is it visible, madam?
Yeah, now first prize.
Yeah. Department of Pharmaceutics oral presentations. First prize goes to Reddivari Harika, Research Scholar from KL University. Congratulations, Harika. And the second prize goes to Mr. C. Mahendranath Reddy, Research Scholar, JSS Academy of Higher Education and Research, Ooty. And from the Department of Pharmacology, first prize goes to Ms. Nazia Begum, Research Scholar, University College of Pharmacy, Osmania University. Nazia, are you there? And
you congratulate her after announcing each.
Yes, madam. Congratulations, Nazia. Second prize in the Department of Pharmacology oral presentations goes to Ms. K. Manipriya, MPharm student, G. Pullareddy College of Pharmacy, Hyderabad. Congratulations, Ms. Manipriya. And coming to the Department of Medicinal Chemistry, first prize goes to Dr. M. T. Sultana, Professor, MNR College of Pharmacy, Hyderabad. Congratulations, madam.
Hello, ma'am. May I audible?
Yes, madam, you're audible.
Yeah, thank you, ma'am, for selecting my research project as a best one. And I appreciate APP organizers and St. Paul College of Pharmacy, all organizing team and the principal sir and the vice principal sir for organizing the conference like this. And a very small time, very short time, your peoples arranged and you conducted the conference in a very good manner. Thank you for your organization and thank you for your prize also, madam. Thank you so much.
Congrats, Dr. Sultana.
Thank you, ma'am.
And the second prize in the Department of Medicinal Chemistry oral presentations goes to Ms. Hina Chowdhury of B.Pharm student of Bharati Vidyapeeth College of Pharmacy. Congratulations, Ms. Hina. Going to the Department of Pharmaceutical Analysis oral presentations, first prize goes to Ms. Preeti Annie Jose, Assistant Professor from MNR College of Pharmacy.
You mentioned university also, Mrs. Harita.
Yes, madam. MNR College of Pharmacy affiliated to Osmania University. Ms. Preeti, are you with us? And second prize in the Department of Pharmaceutical Analysis is for Ms. Sara Afreen, Assistant Professor, Gokaraju Rangaraju College of Pharmacy, affiliated to Osmania University. Congratulations, Ms. Sara Afreen. Are you with us? And we have a consolation prize in the Department of Pharmaceutical Analysis. And the consolation prize goes to Mr. V. Arun Reddy, MPharm student from University College of Technology, Osmania University. Congratulations, Arun. And coming to the Department of Regulatory Affairs, first prize goes to Ms. D. Saujanya, PharmD 4th year student, St. Paul's College of Pharmacy, affiliated to Osmania University. Saujanya, are you with us?
Thank you, ma'am. Thank you for the great opportunity guided by you, ma'am. Thank you very much. Thank you, ma'am.
Congratulations. And the second prize in the Department of Regulatory Affairs goes to Ms. M. Manasa, B.Pharm 3rd year student, St. Paul's College of Pharmacy. Congratulations, Manasa. Are you with us? And coming to the Department of Pharmacy Practice, the first prize goes to Dr. K. Karthikeyan, Professor and Head, Wales Institute of Science and Technology, Chennai. Congratulations, sir. Are you with us? And the second prize goes to Ms. A. Pratibha, PharmD 6th year student, St. Paul's College of Pharmacy. Congratulations, Pratibha. And coming to the Department of Pharmacognosy, first prize goes to Ms. Poonam P. Taru, Vishwakarma University. Congratulations, Ms. Poonam. And the second prize goes to Ms. Uma Rajeshwari Bachu, SRF IICT Tarnaka, Hyderabad. Congratulations, Ms. Uma Rajeshwari. Are you with us? And these are all the prizes for the oral presentations. Congratulations, each and every winner of the awards. And moving on to the poster presentations. In the Department of Pharmaceutics, first prize goes to Dr. Om Bagade, Assistant Professor, Vishwakarma University. Congratulations, Dr. Om Bagade. Are you with us, sir? And moving on to the second prize, it goes to Mr. Sainath Ganoji, PharmD 4th year student, St. Paul's College of Pharmacy. Congratulations, Sainath. Are you with us? And next, going to the Department of Pharmacognosy, first prize goes to Mr. Y. Mahesh, SRM University, Chennai. Congratulations, Mr. Mahesh. And in the Department of Pharmacognosy, second prize poster presentations goes to Mr. Sunil Shevali, Dr. D.Y. Patil Institute of Pharmaceutical Sciences. Congratulations, Mr. Sunil. Are you there with us? And then moving on to the Department of Pharmacology, first prize in the poster presentation goes to Mr. Chenna Anirudh, St. Paul's College of Pharmacy. Congratulations, Anirudh. And the second prize goes to Ms. Gnapika Reddy of St. Paul's College of Pharmacy. Congratulations, Gnapika. And in the Department of Regulatory Affairs, first prize of poster presentations goes to Ms. Akshita Gaud, St. Paul's College of Pharmacy. Congratulations, Akshita. And the second prize goes to Ms. Anusha Talari of St. Paul's College of Pharmacy. Congratulations, Anusha. Are you with us?
Yes, ma'am. Thank you for this prize and opportunity. And thanks to Roja ma'am for guiding me. Thank you so much, ma'am.
Thank you. Congratulations. And in the Department of Pharmacy Practice, first prize in the poster presentation goes to Ms. Suvarna Siddheshwari, PharmD 5th year student of St. Paul's College of Pharmacy. Congratulations, Suvarna. Are you with us? And the second prize goes to Ms. Nausheen of St. Paul's College of Pharmacy. Congratulations, Nausheen. Are you with us?
Nausheen of which year?
She's B Pharmacy, I think, madam.
That's all?
Yes, madam. Once again, hearty congratulations to all the participants and winners of both oral and poster presentations. Now to continue to the program with, a pessimist complains about the wind, an optimist tries to change it, and a leader adjusts the sails. The sails for this particular notion were adjusted by a person who is not only a motivation to all of us, but a visionary and the person who gave us the belief to bring together this event. She is none other than our Vice Principal and convener of today's conference, and I take the pleasure of requesting Dr. Kiranmai Madam to give the conference report. Over to you, Madam.
Thanks, Harita, for your encouraging words. Good afternoon, all. With immense pleasure and happiness, I am Dr. Kiranmai, convener of present conference, is here to present closing remarks of the two-day Indo-US conference hosted by St. Paul's College of Pharmacy in association with Association Professionals Karnataka branch, APP American International branch, APP Analytical branch Chemistry division. As a team, St. Paul's and President of APP, Professor Rajiv Dahiya sir, have started working on this conference in the month of December. Initially,
medicine plant
mute all na once. Initially, it has.
Madam, please unmute yourself, madam, as I've muted everyone. Please unmute.
Yeah.
Yeah, madam. Thank you.
Initially, it has been planned as blended conference, but due to prevailed pandemic situation, 1 week before, organizers have converted to full online mode conference. So I can say it is a little bit easy to conduct offline conferences, physical presence, managing things, organizing multiple tasks, but converting into online suddenly and it's little. I'm not telling that it's difficult task. I never give up in this regard, but it is little difficult. But only with the help of I can say with the scintillating and fantabulous efforts on either side, like St. Paul's and APP, made it possible. So the special thanks and special gratitude to Dr. Rajiv Dahiya sir and our organizing secretary. They have they coordinated with each other and also with the entire team on either side in a very tremendous manner, I can say. It has been truly a great couple of days. Conference has started with a wonderful ceremony, great by Chief Professor Madhav LG, Vice Chancellor Sabarmati University, Gujarat. Department of Pharmacy Sciences, Creighton University, USA. And inauguration remarks were given by Rajiv, Director Faculty of Medical Sciences, University of West Indies, followed by invited lectures by Professor Somnath from Creighton University, USA, and Dr. Gloria. And these sessions, I can say, wonderful sessions, and both the sessions are chaired by Professor S. Lata, Anna University, Professor University, Professor Y. Padmavati, G. Pullareddy of Pharmacy, Usmania University, Dr. Sunita Dahiya, University of Puerto Rico, USA. Both are very informative and thought-provoking sessions, especially scholars oriented. Am I able? No, muted.
Yeah.
Thought-provoking sessions, especially research-oriented gates, enjoyed both the sessions. And brainstorming question and answer session, chairpersons has given notable remarks. And coming to afternoon session of first day, it is really exotic and invincible effort because not many obstacles from set of evaluators due to health issues and some other issues. Last minute, many people dropped because pandemic is very wide transmitted. So by considering all these things, so but St. Paul's team, unbeaten professional attitude, has managed everything perfectly. Excellent presentation from senior scholars across India and oration session hosted by Dr. Rajiv sir. It's really, I enjoyed myself very well. Very informative and very useful, and number is every piece of work they presented is really fantabulous. And everyone truly swimmed across the ocean of research and with respect to trending aspects of pharma research. This session is chaired by Dr. Mehul, Dr. D2 Kataria, Dr. Sunita Dahiya, and Dr. Nilesh Trivedi. So congratulations all. And simultaneously, 12 oral and poster presentation sessions were conducted successfully by moderators of St. Paul's team. And special appreciation to all the moderators of St. Paul's College of Pharmacy, name by name, right from Mrs. Deepika, Associate Professor, Department of Pharmaceutics, Mr. Sudhakar, Associate Professor from Department of Pharmacology, Mr. Moinuddin, Associate Professor from Department of Pharmaceutics, Mr. Santosh, Associate Professor, Department of Pharmacognosy, Mrs. Navya, Associate Professor from Department of Pharmacology, Mr. K. Sarish, Associate Professor from Department of Pharmaceutical Analysis, Mrs. Jagruti, Assistant Professor from Pharmaceutical Chemistry, Dr. KJ Sam from the Department of Pharmacy Practice, and Dr. Samreen Afroj from Department of Pharmacy Practice. Evaluators for all these sessions are from various reputed and premier institutions in and out of India. Name by name, I congratulate all the evaluators for accepting our invitations. Two of them, they deserve a lot of appreciation from organizing committee as they accepted last minute invitations and spent their valuable slots. Name by name, Professor Mazid Iqbal, HOD, MESCO College of Pharmacy, Usmania University, Dr. Mahendran Shekhar, HOD, University of Kuala Lumpur, Malaysia, Dr. Jagadish from MNR College, Dr. G. Sumalata, Associate Professor from Sri Chaitanya Deemed University, Dr. Anusha Joel, Research Scholar from Wales University, Dr. BVS Lakshmi, Cipra Labs Private Limited Team Lead, Dr. D. Prashanti, Associate Professor from Department of Pharmaceutics, G. Pullareddy College of Pharmacy, Usmania University, Dr. J. Archana, Associate Professor from RBVRR, Usmania University, Dr. Shivani Desai, Associate Professor from Dr. D.Y. Patil College of Pharmacy, and Dr. Sneha, HOD, Pharmaceutical Department of St. Paul's College of Pharmacy. So these all acted as evaluators. So we have our heartfelt appreciation to all the evaluators for their kind support, in-time support, and punctuality to the sessions. Oral and poster presentation sessions continued on second day also, second day morning session also. Second day of the conference, so that is today, it has started with inauguration remarks by Dr. Rajiv sir, followed by wonderful scientific lectures by Professor Kuntal Das, Krupanidhi College of Pharmacy, Karnataka, Professor S. Satish Kumar, Wales Deemed to be University, Tamil Nadu, Professor S. Sridevi, Sri Padmavati Mahila Vishwavidyalaya. This session is chaired by Professor Suresh Chennapati, Nalanda College of Pharmacy, Professor Sunil Kumar Chaitanya, HOD, Pharmaceutical Analysis from St. Paul's College of Pharmacy, Professor Kiran Mai, Vice Principal, St. Paul's College of Pharmacy. An intellectual question and answer session is the significant part of every lecture, and everyone thoroughly enjoyed these sessions. So coming to the last part of the wedge of completion of conference, in conclusion, I can say rejuvenate, rigorous, and resonating efforts put forth by Association of Pharmacy Professionals and host institute, that is St. Paul's College of Pharmacy, made this two-day Indo-US conference a very successful event. With the support of, with 500 plus registrations from 45 various organizations across India and 140 scientific abstracts, 24 student and faculty awards, 25 plus presentation awards, and this all would have been impossible without the support and encouragement of management, St. Paul's College of Pharmacy. My heartfelt gratitude to Shri K. Sudhir Garu, Chairman, Correspondent, St. Paul's College of Pharmacy, and Secretary Raghav Reddy Garu, and the entire family of St. Paul's. None is less, none is more. Everyone has put their efforts up to their capacity, and special appreciation as a convener. My special gratitude to Rajiv Dahiya sir and Sunita Dahiya madam, and special appreciation to my colleague HOD, Mr. Somnath Dey, and anchors of 2 days. They put a lot of efforts, Dr. Sneha Thakur. Even vote of thanks is going to be proposed by Somnath Dey. It's my duty to offer special vote of thanks to my anchor team, Dr. Sneha Thakur, Navya, and Mrs. Nagaharita, and many other people over here. Thank you one and all for giving me this opportunity. Thank you Rajiv Dahiya sir to made me national head for Hyderabad chapter, and thank you one and all.
Thank you so much, Madam, for your elaborated report on our conference. Gratitude is the fairest blossom which springs from the soul. Every celebration is an experience. The external fades away, but the internal lingers and echoes in the depth of our hearts. As we come to the close of our valedictory ceremony, I invite Dr. Somnath Dey, HOD, Department of Pharmacology, and Organizing Secretary of today's conference, to propose vote of thanks. Over to you, sir.
Thank you very much, Harita madam. Am I audible? Hello.
Yes, sir.
Yes, you're audible.
I, Dr. Somnath Dey, Organizing Secretary, presenting before you to say good afternoon to everyone, respected dignitaries, invitees, participants, and dear students. I am feeling honored and privileged to get this opportunity to conclude by proposed a vote of thanks of the second International Conference on Recent Advances and Global Trends in Pharmaceutical Sciences. Certainly, these 2 days were very useful, and we all got the opportunity to make aware ourselves of the challenges and development made in the field of global trends and pharmaceutical sciences. Thank you is such a prayer that cannot be seen or touched. It must be felt by the heart. But organizing an international conference is a challenging task. This cannot be possible without great preparation and a wise working hand, support from every corner, and blessings. Now it's time to convey our gratitude to all who have extended their support to make this event successful. At the outset, on behalf of organizing committee, I would like to express the profound gratitude to our beloved chairman and correspondent, also patron of the event, Mr. Sudhir K. Sudhir sir, for his blessings and motivations. Sir, you are our source of motivation and energetic leadership. Thank you, sir, for your blessings always. We most humbly thank you, our secretary, Mr. Raghav Reddy sir, for his exceptional leadership, guidance, and motivation. I'd like to record our deep sense of obligations of the co-patron of this conference and Principal Professor B. Chandrasekhar sir for his motivation and support. Thank you, sir, for shaping us so well. I am happy to offer sincere thanks to our convener, Dr. M. Kiranmai ma'am, Professor and Vice Principal of St. Paul's College of Pharmacy for active guidance and support. Madam, you are given your valuable time in fixing many issues related to this conference. I'd like to thank our co-convener, Dr. Suresh V. Channapati sir, National Head, APP Analytical Chemistry Division, for the various help and we received towards this conference. On behalf of the organizing committee, my sincere thanks goes to our organizing chairman and general secretary of this conference, Dr. Rajiv Dahiya sir and Dr. Sunita Dahiya ma'am, for their wise mentorship and encouragement. Sir and madam are always ready to listen us and immediately ready to solve our problems always when we approach to you. Ladies and gentlemen, our words may not be capable to express our sincere gratitude to our valedictory session guest of honor and President Professor A. Muralidhar Rao sir from APP Telangana State Branch and also Principal St. Mary's College of Pharmacy, Secunderabad. Thank you, sir. I believe this conference has opened a new dimension and our association will go long away. Let me express my deep gratitude to our chief guest, Dr. Madhavai M. Patil, Provost, Sabarmati University, for taking time out for the busy schedule to be with us here today. And sir, your every word is important to us. I convey my sincere thanks to Karnataka State Branch, American International Branch, Analytical Chemistry Divisions, and its official permitted for conducting this such conference. My sincere thanks also go out our university for being this conference to this level. I thank you to our plenary session speakers, Professor Somnath Singh, Professor Sivkanna Fluleria, Professor Kuntal Das, Professor Satish Kumar, Professor A. Sridevi Madam, for their talks, invited speaker and session chairs and delegates who have accepted our invitations and given generously their time to make this conference successful. Your participation, I am sure, has given the depth to the content deliberations. Thank you for your good and humble deliberation towards the delegates. I take this opportunity to thank all the reviewers, technical program committee members for providing their valuable comments in time to help towards the improvement and quality of paper presented at the conference. I also thank authors who have submitted their valuable research to this conference. Equally, on my personal behalf, I am thankful to all the HODs, department assets, my departmental colleagues, coordinators, and organizers for their dedications and support to make it such a great success. I thank personally to the people who work behind the scenes of execute this event, like admin staff, electronic media, technical staff, the stage settings, and other housekeeping staff. Though in the last, but most important, our special thank goes to all the student volunteers who have worked hard successfully for the success of the event. At the end, I bring sincerely apology to those who directly or indirectly help us, but I forgot their name unwillingly. Hope we all will meet our next event shortly. Thank you all. Take care. And at last, first of all, I would like to congratulate APP for choosing me as a active person in National Head in Molecular Pharmacology Division. And I'm assuring you, sir, I will continue this post with your prescribed guidelines, and I have to maintain it in proper manner. Thank you so much, sir. Over to Harita, madam.
Thank you so much, sir. Madam, are we going to end the session?
Yes, sir.
Sir, Rajiv, sir, can we close the session, sir?
So thank you all for your hard effort, and we will meet soon.
Yes, sir. Thank you so much, sir.
Frankly speaking, we won't feel like leaving the platform. Not enough this. So we feel like that. We are not tired.
No, it's good. It was a good effort. Although it is tough for us to organize the 2-day event because for 2 nights, if we are not sleeping, then it's day sleeping. It does not cover well. So, but it's okay. It is we are doing for profession, and we are honoring our professionals. It's good. It gives us a good feeling. We are doing for our profession. So it was a very nice experience to have you all as a good team members, and hope to do some fruitful things for our profession. Thank you.
Yes, sir. Thank you, sir.
Thank you so much, sir.
Okay. See all of you soon.
Yes, sir. Yes, sir.
Thank you so much, sir.
Bye-bye.
Thank you so much.
Thank you.